Okay, everybody, we're going to get started again. All right, so for this next session, Faith is going to be our moderator. So I'll have you come up since you've introduced yourself, you could do that too. And then I have both John and Mooney signed up for the panel discussion after the two presentations. Okay, all right, very good.
Good afternoon, everyone. I'm Faith Bartz-Tarr. I'm with the USAID Bureau for Food Security, and I'll moderate this session on environmental stressors and benefits. The first speaker is Maxine Levin with the USDA Natural Resources Conservation Service, and she'll be talking about soil capabilities to address environmental stressors and benefits.
Okay. So the folks on this list, Steve Monteith is part of your team, your group, and I'm here as his substitute, but also because I'm just, not more than 500 feet in an office that away at the USDA building. But my main topic in my regular work is, as the National Leader for Soil Interpretations, is thinking about soil capability. So I thought I would mention that as opposed to, and then tie it into our soil collection, our soil archive collection, and the potential for research. So let's try to do that. Now, I haven't used this thing here. Is that the way to do it? Yeah. So this is, of course, a table that you can't read. But the most important thing, and you have to remember it's a soil scientist standing up here, is that we've got food security, water security, climate change, biodiversity, ecosystem service, and energy security, but it all ties into soil security in the center. That truly the soils can sit in the middle and be a key or a measure for all of those in different capacities. And since this topic is environmental stressors and benefits, I thought that it would be useful to put this on your radar. It's a paper by McBratney et al., Geoderma, and there have been several conferences specifically on soil security discussing how the topic of soils and the data and the place-based aspect of soils and its capabilities falls within the environmental stressors for a number of subjects and that are important to the environmental health of the world and food security, energy security, all of these different lists. Oops. Oh, what have I got here now? Let's try it. I think I went too far. Let's go back. No, that's it. So I'm going to go back over in a sense, the kind of soil survey products that are that are provided by the soil survey. For the soil survey, it's not only just the U.S., but we have data and collections of data and samples from throughout the world, and we've had those collections for a long time. We also have worked very hard at USDA NRCS Soil Science Division over the last 40 years to work very closely with other countries throughout the world to create databases that are compatible and that feed off of each other and work with each other, and so and that also there are archival collections in other parts of the country of the world that are also building, as well as the soil survey databases. Our soil survey databases, Web Soil Survey, is the access for public and generalized information, but we also have Sergo, which is a much more detailed download of information that can be categorized more specifically. And then GSERGO is this very same data, only gridded. So then in terms of large analysis for the whole country, it can be very quickly done through the gridded SERGO system. Soil interpretations, and this is getting into very specifically what I do and think about what I do, is based on soil properties. And the way the National Cooperative Soil Survey has been done for the last 100 years is that they looked at soil properties and defined soil properties that were stable beyond a 5 to 20 year cycle of use and management. So it's the inherent properties of the soil is what we focused on. And a lot of what food security and environmental stressors now are more interested in, or they think they're more interested in, are soil properties that we call dynamic properties that can be moved very quickly within a year or within a few hours, as some of our biological folks are interested in, that those properties can change very quickly. And the only way we see in terms of a database to formulate how does that work in nature and how can it be applied to other databases and other pieces of information for, let's say, food security or energy security or some of these other securities, is to work through modeling of the inherent properties, and through that make some assumptions. And that's what soil interpretations is all about within the Soil Science Division at NRCS, and that sort of thought pattern. In the past, we've had very broad classes and groups, and I know I put out there for fun our soil planner. I do one every year, a calendar for everybody, for the public. And just this last year, I did it on land capability class, which was the broad-based way that we separated soils into broad classes. And even within in a national sense, when you look at this map that's up there, you get a feeling of where the agriculture is, which is the green, where the class 4 through class 8 lands are in the west, and the mountains of the Appalachians, and the deserts, where you know where agriculture goes and where it doesn't, and where you need potential. The white spots are where we haven't mapped yet in the U.S. We still have some areas that are still not mapped in the U.S. and Alaska, the lower 48 and the upper. So the basic concept, soil capability, and this is for any human use or ecosystem service, it's must be assessed within the context of soil properties, either measured or estimated. And soil capability is what we're thinking about for the context of all agriculture and biological function and ecosystem function. There really is inherently in every soil the potential within the soil properties, the inherent potential to go one way or the other. Lots of times we have a movement these days of soil health, but we think organic carbon, load up that organic carbon. We do that organic carbon, we're going to change the world, and we're going to change those soils permanently. And really, this is one of the concepts that perhaps would bother some people, but it's certainly bothering our agency in terms of messaging. But in the end, the soil only has a certain capability as to how much carbon it can sequester. And no matter how much carbon you dump on, if you don't keep dumping that carbon on every year, in a couple years when you let it go fallow, it will move to its capability, its inherent capability of how far it can go with the system. And in terms of biological response, this is sort of interesting. It gives more credence to thinking through soil properties and their inherent ability, their capability of creating an ecosystem for, let's say, biological root growth, or for soil biology in general, and sometimes even in creating a habitat for biological health and new biology. So within the soil survey, we have thousands of soil series that have been recognized. I think we're up to about 30,000 that are recognized, and they act as reference states for assessment of capability. With the soil collections that we have at Lincoln, and that we actually have small collections throughout the country, in regional and field offices, they help monitor or reference the condition. And so this is the basic concept that we go by when we're thinking about soils and mapping soils and scaling up and scaling down and interpreting all of this information that we have as a baseline information for everyone to use. So the properties that impact soil capability, I know you can't read this, but if you look at the big words, horizon properties, profile properties, and site properties, those are the big pieces that are in the database that are described as place names for all the soils. And so under the horizon properties, I'll go to the profile properties first. Depth to saturation, depth to bedrock, available water storage, depth to a restrictive layer. These are examples. And then site properties, slope gradient, slope shape, slope aspect, precipitation, temperature. These are a day length. These are all aspects of the soil survey that it's data that's attached to the estimated properties or measured properties that are within the soil survey, that each of the maps and the polygons describe the capability of that soil. And then to go, then I'm going to have to, and then horizon properties, even more specific, pH, content of sand, silt, and clay. And for those of you that aren't familiar with soil surveys, soils are naturally horizonated. So you have a surface, you have a subsoil, you have a the contact to bedrock, or never a contact to bedrock, but going within the vadose to a non-biological material. Though these days biological material, we only think about the first 2 meters, but really geographically it goes much deeper and can affect the biology much, much deeper. And so, all of these aspects of horizon properties are in there. And when we're doing interps, we might be cherry picking, or we might be just listing them and looking for indexes, creating indexes that will define a habitat or define the potential for that soil to react in a certain way to either plant growth or to inputs or outputs or soil erosion, various activities. So with our collections, I hate it when somebody gives me a slide and it looks like a normal slide, and then they've got all this little business in there. At the Kellogg Soil Survey Center at the laboratory there in Lincoln, Nebraska, we've got the MIR spectrometer. And this is a perfect example of a technology that we didn't have 20 years ago, that we're now trying to develop and work through. And this spectrometer can measure all sorts of things, essentially just with no contact, where we don't have to digest or destroy the material to actually do an analysis, a chemical analysis of the material. And this is the potential, one of the strong potentials for the collection, because that's one thing I was just asked less than an hour ago, oh, what's your process for access to material? And to tell the truth, I started to think about it, and I think, well, we do have some process because we do lend out material, but the fact is we can't lend it out because whoever works with that material is likely going to permanently destroy it if we lend it out. So, but this is one type of instrument that could be used, and all of this on, to remeasure certain properties. And in fact, what we've done, this is just one example, this MIR mid-range infrared spectrum. We can measure many properties. We have these samples. Where we've already measured chemically total carbon, CEC, clay, pH, carbonates, and now we can go back with this MIR, measure them again, and create calibration curves to measure them other places. And so, the machine is available. We have the reference date. Plus, also, sort of an interesting thing is to go back and remeasure all of the samples and see where there might be differences, and then start reanalyzing the data. That's one of the aspects that is sort of interesting and fun to think about. And the predictions obtained in using these models built from the MI spectra of archived samples is huge. I think there's all sorts of new research questions we could ask that we haven't asked up to this point. And so that's the strength of this collection that we didn't think about before. Like I said earlier yesterday, it was sort of an accidental collection, one of those obsessive things like too much furniture in your house. You just keep collecting and collecting and collecting, thinking you'll use it someday, and then all of a sudden you have a huge collection, and there it is. And now what do I do with it? Do I throw it out or do I keep it? And now I think we have a reason to keep it. So the spectral library, we're calling it a spectral library at this point, as part of our collection, as the baseline for the world, really, in using the spectral information. And so now at this point, I want to just do very quickly interpretive maps, because that's the beauty of the soils collection and the soil survey, is the potential with soil capability to scale up and to create big maps, big data. And, this one is the major land resource stresses, big information. And this is done simply with soil classification, and stretching the data to other databases around the world in soils. Inherent land quality assessment, very similar to the land capability class, only extended out to the whole world. Within the US, we have the National Crop Commodity Potential Index that shows relative productivity under the current climate. This is actually sensitive to other climate. So you can plug in other numbers and you can see where corn's going to be grown if we change the climate, where it'll be most likely better or worse, based on every soil capability, its own individual potential. A very powerful tool. Leaching and runoff combined, we call this the soil vulnerability index. I'm sure there's soil vulnerability indexes that are different if you Google it, but this is ours. And it's mostly unpublished data, and it's used internally through our resource assessment division at the national office for proving that we're making some progress in no-till and other conservation practices for agriculture and stresses. So the benefits of combining soil capability and collections, the main thing is that I'm really thinking the questions of sustainability, climate change at the local, regional, and global scale. And the main purpose is to scale up or scale down. That soil survey in combination with the collection could be very useful. And so I think I'll just leave it at there. We only got 15 minutes. I could talk for hours. So.
Thank you, Maxine. Our next speaker is Edna Makule from the Nelson Mandela African Institute for Science and Technology. She'll be speaking about strategies to reduce post-harvest losses of seasonal fruits in Tanzania.
So, good afternoon, everyone. I feel privileged to stand before you just to present my okay. Good afternoon, everyone. That's okay. Yeah. I feel privileged to stand before you this afternoon just to present my research topic on strategies to reduce post-harvest losses of seasonal fruits in Tanzania. As the world is striving for more food production to sustainably achieve food and nutritional security and more food production to feed the increased population in the future, developing countries like Tanzania are doing the same. In addition to that, is striving to achieve sustainable, site-suitable solution for reduction of post-harvest losses of agricultural produce. Seasonal fruits and vegetables like tomatoes encounter glut harvest in their season. And it is in this time where we experience a lot of losses ranging from 30 to 50%. Therefore, strategies to reduce these losses are inevitable when we want to seriously address the household food and nutrition security. In addressing this, then I will discuss with you a case study of tomato, which was done at our institution with one of our postgraduate students. So, tomato production in Tanzania, we have like 145,000 metric tons, which accounts for about 51% of the total fruits and vegetable production in the country. And the major growing areas are as located with the tomatoes, that's almost around the country with the exception of the middle part where it's a bit drought area. So we all know that tomatoes are potential benefit first nutritional because they provide micronutrients and as well as antioxidant, and for our case it's also economically important because it provides is one of the cash crop for many smallholders farmers and as well as medium scale farmers. So just to give you an overview of the tomato production, the way it increases from 1972 towards, I have data until 2007, but we are still waiting for statistical data from 2009 until 2016. You can also see how we are lagging behind data. So what is the problem then? Approximately 50% of the tomato are lost during the glut harvest season. And there have been several interventions attempting to reduce such huge post-harvest losses, but the moment their impacts have not been realized in Tanzania. And when we try to look on the factors which contribute to these huge post-harvest losses, one of it is limitation to market opportunities. This is due to low quality of the tomato produced in terms of size and grading, and also improper handling at harvest and storage, and also improper storage technologies. We almost have zero storage technologies of our fresh produce. And also packaging in bulk without sorting and grading, inadequate poor processing technologies. There's very pure technologies at the farm level, and also we have poor infrastructures from farm to market. It takes a lot of time to take the fresh produce from the market, from the farm to the market. And it's even worse to the smallholders farmers who are even in the interior, and there is inaccessible roads. They are inaccessible. You can't reach them by roads. So they have to find their own way to market, and that takes a lot of time. And also we have few tomato processing industries in the country which cannot absorb tomatoes harvested during peak season. Currently, we have about 4 big processing plants of tomato, and all these targeted few farmers, and they cannot really reach the smallholder farmers. So these are the pictures trying to address how the situation is, and some of this, the 2 upper pictures we are taking this year, and right now is still the harvesting season, and you can see this is the way. The first picture at the to my side is at the left, is how they harvest and pack in the wooden crates, and the other picture is how they sell it because it's too much and they can't process and they just want to get rid of them. And this is the lower left, is how they transport tomato in bulk. You can imagine transportation like that, how it reaches the market. So the aim of this research work is to introduce optimized site-suitable drying, packaging, and storage technology as the intervention for sustainable reduction of tomato post-harvest losses. When I say site-suitable, it means to come up with the optimal method which will suit the context within Tanzania in terms of infrastructure, in terms of cost effectiveness, and all those things. So this work is divided into several work packages, but today I would like to discuss with you the work package 1, of which is on the progress. And this is development of optimized drying technology for reduction of post-harvest losses of tomato. And this has 4 activities, and the first one is to study the drying performance, trying to compare the drying performance of solar tunnel dryer and electric cabinet dryer. And the second activity was to assess the effect of 2 drying technologies in terms of micronutrient retention of the dried tomato products. And the third one is to evaluate the effect of different treatment on chemical composition of the dried tomato products. And the last one was to assess the shelf life and acceptability of the dried tomato products. So these are the 2 types of dryers we used for the drying. And this is the method. We bought tomatoes from the farmers, we wash them and get rid of debris, and then we sliced them and we dry them using solar tunnel dryer, and the other samples were dried using electric cabinet dryers. And here at the middle, these are the first product, which are the dried tomato slices, and the 2 down there are the tomato powders after pulverizing. So the results show that there was the cabinet The electric cabinet dryer was the most effective in retaining chemical composition. When we say chemical composition here, we mean vitamin C and lycopene. So, as compared to the fresh sample, there were retention in more electric, there was more retention of lycopene and as well as vitamin C for the electric cabinet dryer, probably because it was fast drying and uses shorter time, so there was no time for the, it didn't take long time for the vitamin C to degrade as compared to solar dryer, which took longer time for the drying. And when we checked on the effect of pretreatment on the quality of dried tomato products, we subjected our samples in the 2 treatment, and one with the salt solution, and the other one was the slightly acidic electrolyzed water. And we found that the pretreatment with its salt solution prior drying process was effective in retaining, that was color, vitamin C, and lycopene. So we also further tested on the sensory evaluation, and the results show that the tomato powder pretreated with 2% salt, that was more acceptable, but we also tested shelf life. It didn't took so long, only 3 months, and then it was started deteriorating. Therefore, the availability of various drying method is a great opportunity for the smallholders farmer, which, if grasped with a view of commercialization, can bring substantial reduction of post-harvest losses in tomato. So, what is the future plan? We are still planning to do more optimization on the drying method to attain cost-effective and shelf-stable product, because we find from the preliminary results that the electric cabinet dryer is was the most effective in the drying, but trying to see our local side, we don't have electricity, so we are trying to come up with a kind of design of kind of solar tunnel dryer, but with the properties like those of electric cabinet dryer. And also, we will do field level demonstration of the optimized method and advertisement of the quality dried products for their adoption by smallholder farmers as well as medium-scale farmers. And also, we are planning to do capacity building on smallholders farmers, business skill through training and exposure, just to expose them that this is the way things should be done instead of their ways of doing things. And the 4th one is to promote financial schemes that enable farmers to invest in appropriate irrigation in collaboration with the Department of Sustainable Agriculture at our institution. And lastly, enable farmers group to explore value addition activities through storage and processing. So we are still having some challenges, and while new and improved techniques for extending shelf life of fresh produce exist, reducing post-harvest losses of fresh tomato remains a challenge in Tanzania. And ongoing or new research works and policies are still to provide sustainable solutions through quantifying and characterizing the extent of post-harvest losses across the value chain of tomatoes produced and traded by small-scale producers to determine such suitable priority intervention. It's like we are not really knowing exact magnitude of the post-harvest losses. Some just say 30 and 50, but that is sometimes is too political. It's not depicting what is clearly going underground. So the challenge is now to quantify the post-harvest losses happening until to the level of smallholder farmers. And also the challenge is also to provide necessary technology option for extending the shelf life to satisfy quality and food safety standards. Also building capacity on improving post-harvest handling knowledge to all stakeholders across the tomato value chain, and providing an alternative to dependence on rain-fed farming. As right now, the problem is big because there is a dependence on rain season, that the agriculture section is not like commercialized. So smallholders farmer normally grows during the rain season. That's why we also find tomato, for our case, is also seasonal. And also facilitating formation of strong growers association for easy market access and bargaining power. Just like farmers, when they are in association, then they will have big power to bargain in terms of price, in terms of finding good markets and such thing. So the question is now, which scientific collection can be used and how scientific collections can be used to speed up towards attaining sustainable solution. Apart from working with reduction of post-harvest losses with tomato, we're also trying to reduce post-harvest losses with oranges and mangoes, because these are also a big problem in the country. So, in this slide, I just want to give you an overview of where is NMIST. NMIST is Nelson Mandela African Institute of Science and Technology. We are situated in Arusha, and you can find us in the given website below, and our motto is Academia for Science for Society, meaning that we are trying to work to do research which will provide solution to the community surround us. And I'm working with at the Department of Food Biotechnology and Nutritional Sciences, which is among the department found in Nelson Mandela, and our research priorities are, the first one is what I've discussed here, is on post-harvest management of food, and we mainly focus on reduction of post-harvest losses for improved food and nutritional security. And currently we are working on maize, legumes, and seasonal fruits. We are also working on value addition, and that's product development and quality improvement of existing product. And we are currently working on cashew apples and tomato and sesame. And another area of our research interest is food safety, on which we are our focus on the physical, chemical, and microbiological safety of food. And currently there is a project going on arsenic contamination on rice to set standard for codex, and also the reduction of exposure on mycotoxin, reduction of mycotoxin exposure to human. This is mainly on maize, and yeah, we are having a project which is funded by Bill and Melinda Gates on this. And we are also working on functional foods, whereby our focus is on edible plant with medicinal effect, trying to investigate on bioactive compounds from foods. And the last is on nutritional survey and the intervention, whereby the main focus is on food quality and quantity for health of people, fortification, and supplementation. So, thank you for your kind attention, and welcome to NMIST. I would like to acknowledge the Government of United Republic of Tanzania through Commission for Science and Technology for financial assistance on this tomato research. Thank you.
Okay, so I'm testing.
Yep.
Can you hear me?
Sounds good.
Okay.
Will you introduce yourself?
I'm John Dickey. I'm Assistant Head of Collections at the Royal Botanic Gardens, Kew. My particular responsibility is for the seed collections at the Millennium Seed Bank and for the DNA and tissue collections at Kew itself. Thank you.
Hi, I'm Stephanie Yarwood. I'm a soil microbial ecologist at University of Maryland, and we maintain a small collection ourselves, but we most recently have National Science Foundation funding to use the NEON soil samples that are being housed at Argonne National Labs and to develop some new methods to use extracellular DNA, maybe to track earthworms using those samples.
My name is Muni Muniappan. I'm the director of the Integrated Pest Management Collaborative Research Support Program. Now it is called as the IPM Innovation Lab. It is a USAID funded project, and we are working currently in 7 countries, 4 in Asia and 3 in Africa. The Asian countries are Bangladesh, Nepal, Vietnam, and Cambodia. And in Africa, we work in Kenya, Ethiopia, and Tanzania. In the past, we have been concentrating on developing IPM packages for high-value vegetable crops. Currently, we expanded that to fruit crops as well as cereals.
Thanks. So I guess first I can open it up to the panelists if there's anything any of you had in terms of feedback about the presentations or questions or comments you wanted to make about the role of scientific collections to address environmental stressors and benefits. Did any of you have something you wanted to?
I had maybe a question or a comment for Maxine about the NRCS samples, and I guess I'm, as a soil scientist, aware of the soil survey, but didn't think about the fact you guys had soils necessarily. But I wondered about storage conditions for those soils. And my guess is that they're stored maybe dry, for instance, or, and I think that one of the challenges we maybe have in soils is that if we're going to do any kind of biological kinds of work on those, we want them like at -80, but there's lots of issues with power and things like that. And I wondered kind of your comments or thoughts on that.
Okay. Yeah, and it's interesting that you're working on the NEON because NRCS collected every single one of those samples for you.
Yeah.
So, and in the case of the biological elements, we tried to keep a moist sample and send it directly, but we didn't save this moist sample in the facility in Lincoln because all of our samples are dried to 37 degrees centigrade for 48 hours. And then we have 2 samples with every horizon, which is, one is what we call natural fabric that keeps somewhat, as it came in the bag, the natural structure and porosity. And then the other sample is ground and sieved to a 2 millimeter sieve. And then, of course, both samples are dried for 48 hours and kept dry. The facility itself is the basement, 15,000 square feet, and kept in small cardboard containers. There's nothing hermetically sealed about them, and that the temperature is what the temperature is in the facility. And I think you might have saw the pictures if you saw yesterday, but, funky plastic over the tops just in case we have some sort of fire or something and the sprinklers come on, and at least they won't get the samples too wet. So, for biological, that's the negative aspect of that. If you're trying to work actively with biological samples and hoping for some kind of miracle, that our 1940 samples might still have residues of earthworm activity that you can measure within our samples. I don't know. I don't think so. But if you look at the other aspects of the strength of what we can provide in terms of inherent soil properties that create a framework or a habitat for microbiological, because the majority of the microbiological activity happens within the pores. And so if you have a measure of the porosity, you can start to define where you'll see those samples or not. And CDC has come to us, the interpretation staff, several times, and we've created large nationwide maps for specific organisms of where they'll likely show up. And they are told, you know, when we make those maps independently based on what we think will be the characteristics that those microorganisms would prefer within the capability of each soil, we show them the maps and they go, OMG, that's exactly, you know, where we're seeing these areas. And they say, give me those maps. And then some of them have actually been for some actual disease, and top secret, because they don't want people to know how dangerously close we are to annihilation on some organisms. So I think that's where the power of the collections are. Something I didn't spend any time talking about was in the process of getting ready for this little conference, you know, we had lots of discussions of, you know, what else can we do with these samples? And I made little questions, you know, like, you know, besides just the MIR standardization of a new technique of sampling and analyzing and using it as a reference, are there other analyses that we can do now that we just didn't think about before? And I brought up pesticide residue. You know, we have samples from 1940. Every year, more samples were collected. Maybe we can do an analysis looking to see, search for residues. You know, we've never measured that, but, and then with the new MIR, we probably can measure it very quickly and easily. And also some of the other tools that are non-contact, non-invasive, where we're not destroying the sample and making the analysis. So there's a lot of interesting questions there that we haven't explored, but I think we should, as well as mining the database for organic carbon and through the years, things like that.
I have a comment for Edna. You know, we work in Tanzania in developing IPM packages for vegetable crops. You know, in 2006, a pest called South American tomato leaf miner, scientific name is Tuta absoluta, got introduced to Spain. And it has covered the whole of Europe by 2008, and it crossed the Mediterranean by 2010. It reached Tanzania in 2014. So if no control measures are taken up, it will wipe out the whole crop. Now the farmers are spraying different kinds of pesticides, so they may get about 50% yield. So if the pre-harvest control measures are not taken up, it is going to cause a very serious problem. So in my project, I primarily use the museum collections as well as the taxonomic expertise in getting these specimens identified and also surveying for natural enemies, sending those natural enemy specimens to this museum and getting them identified. So eventually, if we find an effective silver bullet, we would be able to use that. We did use in the case of papaya mealybug. That also got introduced recently into Tanzania. It is causing serious problem. I've been approaching the Tanzanian government as well as USAID mission to take up the biological control program, but I wasn't getting any response back. But this project was taken up in India, Sri Lanka, and other countries, and it has given very good results. Our economists came up with a cost-benefit analysis of just introducing this classical biological control of papaya mealybug in India alone. India benefited anywhere from 500 million to 1.34 billion.
Actually, it's okay. Okay, there is we have heard about the tuta absoluta disease, and there is one PhD student who is currently working on mapping the exactly area where this disease has affected, and thereafter she's supposed to come out with the possible solution on how to prevent it and to mitigate so that it doesn't spread. So I don't know how far we will reach towards obtaining the solution because we don't know exactly now where are the specific areas, I mean, where are the affected areas currently?
Well, this pest, moved in from Spain to Europe and then to East Africa through Sudan, and then it has come down to Tanzania in 2014. Right now, it covered Tanzania and it has already moved into Zambia. So it is already there. There's quite a bit of work has been done on it in Europe and other places. In fact, we will have a special symposium next Tuesday at the International Congress of Entomology in Orlando, Florida.
Are there data showing the specific areas affected by the tuta absoluta in Tanzania?
Everywhere, wherever tomatoes are grown in Tanzania, affected by tuta.
I want to go back. Well, first, a short question for the tomatoes in Tanzania. And it's nothing to do with collections, really. Why not pulp them and can them rather than fret about drying them? Pulp them and can them.
Yes, that could be the good idea to can them. But the problem is coming that we are targeting the smallholder farmers who can't even supply to the industries for processing and those kind of stuff. But also our industries, we have about 4 tomato processing industry. They are mainly produce tomato paste and ketchups, but they haven't gone for canning, and I think it's also a point of marketing. Yeah, there's no market for it, and they can't also export it because of the standards. It doesn't meet the standards.
Yeah.
So we are trying to target the local market for the moment.
Because my imagination struggled a little bit with how collections could help you with your problem, but I was sort of going down the path of, are there tomato varieties that are tougher, that take the handling that perhaps you're not using, so you'd be into the international databases for because it's an important crop, or maybe there's something residing in wild Solanaceae that the breeders could get across, but that the time scale on that's going to be it's out of sync. It's not going to solve your problem quickly, is it?
Yeah, that's also part of our challenge. We can't get the real solution to solve the problem quickly. So it's a kind of trying and error, but finally we will reach to a solution, I hope.
Can I have another go? It's about soils, back to Maxine. So it sounds impressive how you can do predictive modeling based on soil physical properties as that's a matrix for the microbiological population. I mean, the people are telling you, crikey, you got it right. That I hadn't spotted that before. So are you able to model macronutrient availability then? Nitrogen, phosphorus?
I would say potentially. Maybe it's beyond my small little staff, but I think that's an aspect of working with the power of the soil survey to work forward. And a good deal of the models out there are starting to make use of that. And that's the way the models are working. Up till this point, a lot of the models have focused on soil erosion and that physicality, but I believe that they can also start working towards other aspects, more biological aspects in the approach of habitat, you know, of defining the habitat down to the micro level of porosity, you know, measuring the pores, thinking through those pieces. And all of those pieces are potentially in the soil survey database and their potential. Something that I was just thinking about the collections, thinking about the MIR, the DNIR, the very near infrared, and also X-ray fluorescence. There's all these different sort of non-contact measurements. And also looking at EC, electrical conductivity with the electromagnetic induction instruments. And I just gave a talk yesterday with Chinese. We had a Chinese delegation downtown, and in the very slow dialogue that we had with translation, they sort of had an epiphany with precision farming and these non-contact instruments. Could we measure contamination as you go, as if in prediction in farming, in precision farming, and then create solutions that way? And you know, their big problem is contamination of big systems, perhaps also in Brazil and other places where we've got big agriculture that kind of made big mistakes in large areas, or in reclamation, even in small areas. And I thought, you know, with the collections, what would be very interesting in tying precision farming to the collections is to start doing these MIR reference bases of the plant material itself, and making references, also measurements of contaminants or pesticides, pesticide residues. So then in a precision farming, you might be going over a space with these measurements and comparing them from the reference states. It's just a thought. I mean, just tying together what I talked about with the Chinese for a very short period yesterday, and then, or the day before, and then, no, it was yesterday. Time goes by fast. And in the morning, and then coming to this conference in the afternoon. I think there's a lot of power in some of these non-contact measurements, the collections, and creating reference basis for measurements.
Well, I think we have maybe time for one more question from the panel itself, and then open it up to the rest of the group. I think it would be nice to tie in the aspect of the environmental stressors by asking if any of the panelists have had experience combining data from weather services with data from collections to address research questions, and what their experiences have been related to that.
Kind of. We use WorldClim for global macroclimate data to try and predict the optimum conditions for germination of all our collections, because it's a global collection. They come from all over the place, and we struggle with wild species frequently to try and assess what are the best conditions to get maximum germination of these wild species, which are often riddled with quite deep dormancy. So we're having quite a bit of success with just following, not microclimate, because we can't get it obviously, but using macroclimate, seasonal temperature changes, for instance, to get successful germination of our collections as the assessment of viability and making them available for use. Yeah, we do.
Yeah, we've actually done quite a bit of work here in Maryland over the last several years looking at greenhouse gas emissions, so N2O, out of agricultural crops. And, it's, we've done everything from National Weather Service all the way down to our own rain gauges and things like that to try to predict it. And it's complicated. And one of the things that we're finding challenging is as we adopt new technologies in agriculture, such as cover cropping, we have, the same types of soils and the same microorganisms, but we deal with, dormancy and, very small differences in scale, making a very big difference in terms of the activity of those organisms. And so it's been challenging to integrate that.
We have a project in Nepal to study the climate change and biodiversity. What we found over there is, the climate difference over there, what we can get from Florida Keys all the way up to Arctic, we can find that much change within 180 kilometers in Nepal. So we have set up a project to set up weather stations every 500-meter altitude, starting from the Terai, that is almost at the sea level, up to about 3,000 meters. And in all these 500-meter altitude, we set up a weather station and also assess the biodiversity and see as the climate change changes, progresses upwards, whether there is any change in the biodiversity. And also we are checking on with the remote sensing photographs that have been taken last 20 years to see any of these invasive species like Chromalina has moved from lower elevation to higher elevations. And similarly, even we are looking for other plant biota, see whether there is any movement that direction.
So I gave the example of the NCCPI, that model that we have, which is very climate sensitive in showing crop commodities and their potential productivity index for the U.S. It's a simple model, so it could be applied anywhere where there's a soils database. All of the soils databases for the soil survey in the U.S. are tied to climate as we saw it when we were, as the data was available during the period of time that we mapped those soils. We're now wanting to go back and take more robust data on climate, including microclimate, and apply it to the soil survey, the physicality of the soil, and then rethink the database itself and some of our models and think through that, whether we can fine tune our information. As it applies to the collection, those are different questions in terms of the scientific merit and thinking that through, but I think that's where we can go, is we can start thinking through that collection and how we can exchange time for space and space for time in terms of thinking through what we have then and what we have now. I think there's a lot of scientific questions we could think through and answer with those, with that collection that we just haven't thought about before.
And I think it's a good time to open it up to the group. It seems like.
The data that you showed, a list of data fields for the collection, included precipitation. Do you know how that's determined? Is that like a rain gauge in the field of the sample, or is it something more general?
No, it's much more general. It's based on 30 years of rain collection at the time that we did the soil survey. And so that's why lots of times we think it would be nice to restructure that data to something more current, because many times, it was a collection from 1950 to 1980. And that was the time of the soil survey, so we went back 30 years, and then we established our rainfall precipitation as a range in the database. And now we have so much better data to work with. and that's one way that I think we can separate. But we can do that with the soil survey, is that we can just remove that aspect of the database that's in the soil survey now and overlay it with new spatially defined information and see if we get different answers. And retrospectively, yeah, and go back to local records, or just, there's a lot of power in playing with the data.
So I'm always amazed that it seems like the soil microenvironment is just the most complex thing one gets studied, but it's also an area where there's the most overlap with other collections, and the microbiome project is one of the ones that is coming to the fore. And I'm wondering I want a clarification on sort of experimental model methodologies. So with a cow, right, we have rumen and we have fistulated cows, so we can go in and take samples, but we got the cow running around. So in the sense of the cow, it's the whole ecosystem with regards to the soil. So you can have plots in the ground, whatever you can do, take samples. But in the laboratory, they've developed an artificial gut for a cow, and they can actually put it on the wall and they can study the movement of material through the gut. Is there anything in a soil where you can keep it in the lab? I wasn't clear about whether there's the equivalent of a fistulated soil in a lab, so that you could go in and maintain essentially the biological integrity of the soil for long periods of time, and there are collections like that.
You probably have a better answer.
Actually, there might be. So we actually have a project in my research group working with the Naval Research Lab where they're actually using a technology called soil printing. And the idea of soil printing first is to take and use soil essentially like you would print cells and actually knock pieces of soil off and be able to culture those microorganisms. The second part of that project is to actually try to construct soil. And so it's actually to design a printer that basically mimics texture and macronutrients. And so essentially to try to put that together. There's lots of technical issues with that that we've discovered, as you might imagine. But that's exactly what we're trying to do for that very reason, because, if we're really going to mechanistically test it, we have to be able to recreate it. And so maybe it'll work. But as far as I know, that's the only project that's doing that. A few other people have attempted that kind of same thing, but no one's really done it successfully. Printer. Yeah. So the idea of soil printing is that, laser printing essentially doesn't, it uses a laser to take ink, right? And to drop that onto a piece of paper. The idea here is to actually use lasers. They've actually done this with cells actually for quite some time now, but essentially to take the laser, create a ribbon of soil and to knock little pieces of that off, and to be able to then culture and use that independently. So, so it's printer. Printing. Yeah. My collaborator at the National Naval Lab is actually published on that.
And then we don't do it at the National Soil Survey Center or the laboratory in Lincoln, Nebraska, but some of the scientists around the country in soil physics have had some fun with taking an MRI of soil samples and creating slices of porosity, and then essentially like a printer, definitely measuring the porosity through that methodology. And I think that's kind of fun. I would like to continue looking at that. 30 years ago, when I was working at Oak Ridge National Lab as a technician, we would do little microcosms, little soil cores, and grow stuff in them, and do very infinitesimal measurements on water movement with pore microtubes, and have it in glass containers and try to look and see what we were doing. I don't know. I haven't tracked whether anybody ever repeated any of those experiments. but there they are. But I think that once again that there's potential. It would be fun to take our soils collection and do an MRI on the natural fabric and set up a standard of porosity as measured that way, as opposed to other ways, or doing a print. So we start having some generalized different kinds of measurements that then can be plugged into models and described and use them as a reference base. But, trying to be expansive.
By curiosity, would there be any comparative advantage in establishing a collection of frozen samples of soil, the comparable system of what they have established as, let's say, core of ice from polar areas, as compared to what you have already established?
Yeah, I would love to see that. We haven't done it. I think the Europeans have done it some, where they've made cores. That's what I think. I'd have to check it out. But, oh, did he? Did he? Well, they've never showed them to me, but, and I don't know where the data lies in the database, but we don't all talk to each other every moment, and I'm not as familiar, but I would expect that. Something else that we have is that we have collected soil monoliths, and all the universities around the country have done soil monoliths where it's a slice of the soil. They dig a pit and they do a slice of it, and then they impregnate it with wax or glue. And if some of you had ever been to the Smithsonian for the Dig It exhibit, we had all 50 states and actually 52 monoliths of the state soils and territory soils for the U.S. Now, that wasn't a scientific collection. It was more just a public information collection that showed people how soils had different structure and different texture and different colors, and gave people some ownership of their soil in their state, what it might look like. And it's only impressive when you have them all there. When you just one, it's like, not that interesting. But when there's 50 of them, then you get kind of excited. I think the core idea is very good, just like the ice cores from the Arctic, just like you said. And to maintain those and keep those, it might be, if nothing else, because porosity and structure is so much more important now, I think, than now that we're thinking about biologically, than it was before, when we originally started collecting these samples. So it'd be good.
With the ice, with the frozen soil cores, if you want to do the biological side of that, you might have a problem, because there'll be differential survival of freezing, whether organisms are spore formers or what. There's quite a bit of so you probably won't get the biological population that you had in fresh soil.
Yeah, so the freezing, and I don't think, at least in Lincoln, we won't have the capacity to do that. We're only just now in Lincoln, Nebraska, with that laboratory starting to think about the biological side of things and what is possible and what we can do. And whether there's other places in the country that could do it better than us. I don't know.
This is more of a comment for Edna. 100 years ago, the United States had significant problems with tomatoes. All the same problems that you're encountering in Tanzania with post-harvest losses. What might be project is to look, the United States tomato growing fields eventually moved to where they are in the United States, sort of by hit and miss because of regional problems. They moved from Ohio to Indiana, eventually out to California due to climatic issues. It might be possible to look at the movement of tomato growing in the United States with weather and soil fungi, and sort of match regions in the United States to Tanzania to determine which would be the optimal areas for growing tomatoes and what would be the suboptimal areas. That'd be one way to tie in weather and soil fungi and everything else, because 100 years ago we had a horrendous problem here.
It's a good idea that we can look to what happened to United States and try to apply it in Africa. Is that what you suggest? But sometimes there are challenges, because copying straight from United States and implement it in Africa, it might have it can be a kind of solution for a short time, but it can't be sustainable. To my way of thinking is we can start from identify the problems, our problems, and then solving them into African context.
Thank you. Thank you for getting back on this topic. I've been hoping to get back to the tomato question. The immediate past president of the National Association of Plant Breeders in the US is the tomato breeder in Ohio. So just to underscore that scientific collections with genetic diversity of tomatoes can allow you to do things that otherwise you couldn't do. And if I could just make a suggestion for you when you go back home, and I'm speaking to Makule. It's Makule. When you go home, 20 years ago maybe, I worked with the Tanzanian National Program on cassava breeding. So I know that there are good plant breeders in Tanzania. And the World Vegetable Center is aware of Tanzanian situation. And so at the same time that you're looking at processing, that would be surely an interesting and rewarding thing for your organization to do, to get in touch with your national plant breeders and ask them, do they have the opportunity to work with the same farmers to bring in some of those scientifically collected tomato varieties, a scientific collection of world tomato varieties, and maybe see how they interact with the local strains, and maybe you can get something that uses your methods of drying, but maybe even faster or more reliably or with higher yield. That would surely, well, you may be doing it already, but if you're not, you probably could make some interesting gains pretty quickly, and it would be quite rewarding, I would think. Thank you.
Yeah, the breeding program on tomato is there, but the problem we are trying to address, because I'm the food technologist, so we are trying to take from the harvesting point what's happening there. It's not like there is no breeding, but it's where to sell, how to prolong the shelf life of the produce. Yeah, because it's too much and a lot is waste. That's the problem. But the problem, probably breeding can also help just to improve the quality so that it can expand the market. Probably even export, something like that. But what we are crying right now is how can we reduce the losses, because it's too much.
Yeah, I want to follow up on that. And I'm assuming from what you've said that the post-harvest problem is not any pest. It's not that that the tomatoes aren't being attacked. It's just that it takes too long and too much handling to get to market. So I want to reverse the question and go back to what we learned what we discussed yesterday about traditional knowledge. Tanzania is not a small country, so I'm assuming that tomato growing is going on. Your map showed it was going on all around the country. I'm assuming it's different habitats, different microclimates, different substrates. How much specialization has there been of different tomato strains by local farmers to match local conditions? I mean, how much variation is there among the local strains of tomato that are being grown in Tanzania, and how much interchange, how much crossbreeding, how much backing up of the tomato crops is going on? Or are the local, the small growers, are they really very isolated and not communicating with each other? Because it sounds like it could be a very interesting natural experiment, a natural laboratory for the use of local variations in different habitats.
If I get your question correct, it's like how much the local varieties are used by the small farmers. Based on my experience, is for the small holder farmers, they normally use the local varieties. They don't go for the new, the modern cyst for the tomato. So it's kind of, they grow what they, sometimes after harvesting, they prepare their own seeds, and that's what they produce. And sometimes I think there is even no connections between them. So what is situated, let's say, in the eastern part can remain eastern if there is no kind of research or institution taking from one place to another. But between them, there is no communication. It's kind of, yeah.
Yeah, that's too bad, but maybe that's one solution. I might get in trouble for saying this by some people, but I wouldn't recommend necessarily following the American model, because our breeders forgot about taste when they switched to producing fruits that were more easily harvested and more easily stored. So just remind your breeders to keep in mind the taste of the product, because the American tomato has gone downhill in quality, in my opinion. But I believe there's some literature out there about the time of day when the tomatoes are harvested, like how hot it is, harvesting early in the morning instead of in the middle of the afternoon. That could help with the storage. And also insect damage. We can improve storage by reducing insect damage to the fruit. Just a couple ideas.
Yeah, I agree with you. We can adopt the American ways, but then it really needs time. It can't penetrate the way it is. It has to get adopted first by farmers, and sometimes, yeah, I'm still thinking it can be, maybe partly can be absorbed and partly has to take the basics of the, yeah, African way.
I used to consult a little bit for some Tanzanian farmers' rights organizations, so I can say the agricultural system in Tanzania is pretty sophisticated. There's not a whole lot of knowledge I think that we could really bring, honestly, certainly in this group. But you brought up something that's very important, I think, for us to consider. So in many countries throughout the world, the resource-poor farmers are dependent on open pollinated varieties or true breeding varieties, which is what you just described. They're saving their own seed, and so the economics of growing F1 hybrid seed are not really workable in those environments. And those represent that production model represents a very different collections strategy and breeding improvement strategy in some cases than does the kind of breeding that a lot of us automatically think about when we're thinking about plant resources that are eventually going to go into breeding programs, which is you provide the genes into some very sophisticated, often commercial or university or Department of Agriculture sponsored breeding program that's going to integrate the genes, turn them into F1 into inbred lines, and then and then there's a sophisticated seed distribution system out there that can sell and often certify the quality of those seeds. So I think it's really important for us to recognize that there's a different model that's very, very important, especially in the developing world. And for those that do work in the developing world, I mean, this is UNFAO and CGIAR and the Gates Foundation and everybody else that deals with this stuff. Has oftentimes two completely separate pipelines. For those. I think the most probably obvious or famous example, at least to Western groups, would be the WEMA project, Water Efficient Maize for Africa. Tanzania is a big location for testing of WEMA, and they're simultaneously breeding true breeding varieties, open pollinated varieties, F1 hybrids, using GMOs in some cases, non-traditional breeding, marker system breeding. They're really doing all of those because no one solution will work for all the farmers, depending on the resource capacity of the farmers, their distance from market. And so the germplasm collection folks that are feeding into those groups really have to have all that in mind completely up front. There's also been some really fascinating inclusive decision-making analysis done by Tanzanian researchers related to the WEMA project about how the participatory element of getting the end users, the farmers involved in upfront, before you even start breeding, so at the collection strategy, how that is really critical for building, not only the proper, understanding what the production models are going to be that the farmers are going to employ, what their capacity is, what the infrastructure can support, but also building trust, and especially where non-local players get involved. Large international development organizations, scientists with the best intentions from other countries. Trust building is, often ends up being a major linchpin. So there's actually a pretty robust literature on this topic, and Tanzania is actually one of the epicenters of where this is being studied and worked on in multiple different crops. And I think it's an important thing for us to consider as we think about collections policies and management and data sharing, because as we alluded to earlier on, things like just the cultural practices in living collections for how you keep something alive, whether it's a fungus or a plant or whatever, that's not being captured in our metadata, but neither is the preferences of the farmers, the relationships that they have with various organizations in food security, and how that should be informing the most efficient usage and development of germplasm and other collections priorities and systems.
You're welcome.
Well, I was just thinking it's really too bad the farmers don't talk to each other more, because one of the things that's been effective with some crops is setting up seed exchange networks. And I'm just thinking, what if the perfect drying tomato is out there somewhere being grown, that is well suited to the kind of processing that you want to do? It could be out there and you would not necessarily know. And even though I know you just work with the processing end of things, there could be some real benefits to connecting with the farmers who grow these traditional varieties. And with them being in touch with each other, then they get together and exchange seeds and information. And that can really help preserve biodiversity. In a number of crops, people have done things like that.
Yeah.
One last question before we move on to the next item.
Makulda,
If I understood correctly, is this tomato targeted towards local market?
They are traded in the local market.
Okay. So before your research started, did you try to survey any existing traditional processing methods that were available?
Sorry, I don't get it.
At any point in your project, did you try to find out any existing traditional tomato processing techniques or methods available?
You mean that if there are traditional
Yeah.
local processing available? Currently, they are trying. There is a way they are drying tomatoes. They slice them and dry it on the ground. So the safety part of it is questionable.
Okay,
That's
but
the
are
only
you
way.
aware that there are other traditional processing technologies that might fit into low tech, low energy processing technologies that might fit in your case?
In this case, the traditional, you mean the traditional method which can fit in?
Yeah, from maybe from other parts of Tanzania, or maybe from other countries in Eastern Africa, or maybe from West Africa.
Sorry, I don't get it. Your question. Other ways of drying tomatoes. I think there should be other ways of drying tomatoes from other countries, but what we are trying to really focus on is to get the cost-effective method, like a kind of the method which will be affordable and reliable, which it doesn't cost so much for the small holder farmers to do it. That's why we opt for drying. We couldn't go for the expensive technologies, and further to this is like of the where the smallholders farmers exist is a kind of remote, and the access to energy is almost zero. So using solar energy could be an option.
Well, I'm aware that in Ghana, people pulp tomato, and then they put them in glass containers, and they try to pasteurize them, and they can stay for some time, which could find them be useful in the local market. And I didn't know if you tried to have any collaboration with the polymer science departments of your university? Your universities, I mean.
Yeah, when you are saying trying to pasteurize tomato in containers, that's you are using energy. We are trying to find the cheapest way to really address this.
Yeah, but you couldn't tell
low
that
income
it's
community.
actually expensive, right?
Sorry?
I mean, you couldn't say that the other solutions in other parts of Africa might be expensive when you've not known them, could you?
I could say other solutions, they are expensive.
What I'm trying to say is that there might be cheaper solutions out there, and you need to look for them.
Yeah, that's why we are researching. We haven't got to the conclusion. We might find
Beyond
that.
Tanzania, maybe.
Sorry?
And you need to talk to the material scientist.
To the material scientist. Okay, thank you.
Okay. All right. Thanks, everyone. Okay, so it's 3 o'clock or so. We're actually going to start the next session a little bit early. So if we can be back at 3:15, and we'll get started again then. Okay, thank you.
Is the mic on? Is it on? I can't hear it, but that's not what's important. What's important is if other people can hear it. Megan, USDA has the Ag Research Service in the building just over the other side of that campus. Yeah, they have an actual program leader for agriculture. Well, but I don't know that ARs work on coaching. They have done on that. We'll go back and forth between the saltwater. I know that when I was thinking that you were something completely different, but actually there's a link there. Maybe we'll do something about it. Can you talk? I think that we have yeah. Do you want me to start calling people up in, or do you want to start calling? Ladies and gentlemen, come on in. Let's hear our colleague from Florida Atlantic University who has to leave soon. So we want to be sure and hear what she has to share with us. Bring your coffee, bring your snacks, and you can't bring your conversation, I'm afraid. But are we, Eileen, does that mean we'll be finishing 15 minutes early so people could continue their conversations at that time, or we'll just have a longer time for discussion later? If you could give me the signal when you think, go now. Okay. All right. We are about to begin. Good afternoon, and may I introduce myself? My name is Anne Marie Throw, and I am in USDA. I'm one of the worker bees in USDA. Maybe not. Not sure if I pollinate, but I certainly buzz around and try to help make things happen. And I wonder, those of you that are our guests, that are our visitors, you may or may not know about the founding of USDA. It was founded in 1862 by Abraham Lincoln, after whom this building is named. That was still during the Civil War. But Lincoln and his Congress already knew that the agricultural soils west of, or the fertile soils west of the Mississippi would be opening up to agriculture. So there would be a demand for knowledge about agriculture. And of course, Lincoln himself was from Illinois, which remains a very productive agricultural state. And in the initial act of Congress that set up USDA, it had two main components of its mission. One was seeds and the other was information. So that's very much relevant to what we're talking about today. And they both remain core elements of USDA's research mission. Seeds through the National Plant Germplasm System that we heard about yesterday, and of course through breeding and genomics program, and then information here embodied in the National Ag Library. So what we're doing here today is very much in line with the 150-year mission of USDA. And our first speaker then is Megan Davis. Dr. Davis comes to us from Florida Atlantic University with a PhD in marine science, and she's going to speak to us about aquaculture and food security. She's going to start off our session, and then we'll immediately have some question time, and then so that we can talk to her a bit before she leaves, and then we'll get on with the rest of the panel.
Okay. Thank you, Anne Marie. I also want to thank Eileen for inviting me to this great session. Okay. And for USDA for being the host. I've certainly enjoyed the breadth and scope of this meeting. I've learned so much. I'm going to take you to the other part of the planet, to the 70% of the ocean, the ocean part, the water part of the world. And we're going to talk a little bit about the oceans, fisheries, aquaculture, also known as fish farming. And so I'm going to move on from there. First of all, I just want to engage you and ask you, how many of you eat seafood? Almost everybody. How many of you eat a lot of seafood? Okay, well, definitely seafood is on an all-time high right now. Worldwide, it is about 16% of the protein that's consumed. In the United States, it's closer to about 6%. And you can see here that the amount of seafood has certainly increased over time due to mostly improved distribution and access to seafood, but also because people are becoming more and more aware of the nutritional value of seafood. And you can see over the last almost 6 decades that the consumption of seafood has gone from about 10 kilograms per person per year to now 19 kilograms, which is close to 40 pounds of seafood per year per person. So it's definitely increased in some countries in Africa and also in Asia. The seafood consumption is double that. It's about 50% of the protein intake. And so I also put down the bottom here with global population at about 3 billion in 1960 and 7 billion in 2011. So there's an issue right now with seafood. As many of you might know, much of the world's fisheries is becoming overfished. And you can see here that about 53% of the fisheries is fully exploited and 32% is overexploited, whereas there's about 10% of the fisheries, 10 or 15% of the fisheries that hasn't, it's being fished sustainably or not at all. And so this is really a big issue. And I don't know if you've thought much about this, but I think about this quite a lot, that the fisheries, the seafood that we're taking from the ocean, the wild seafood, is really the last frontier of a commercial protein source in our world. And so it is becoming overexploited. It's pretty much plateaued in the amount of seafood and protein that we can take from the ocean. So with this pressure on the wild stocks, let's talk about where the food is actually coming from, where the seafood's coming from. So FAO puts out every 2 years a report on the state of aquaculture and fisheries. And this is the last, from their last report in 2014. And you can see here that you have fisheries, you have aquaculture, you have about 158 million tons of seafood combined for 2012. That's a lot of seafood. I always, when I'm teaching this, I talk about 1 ton being the equivalent of a Volkswagen. So that's like 158 million Volkswagens versus seafood coming from both the ocean and aquaculture. Just to give you some context, that's a lot of seafood. And what's very interesting to me when I look at this, when you look at the inland portion here, the inland portion is mostly your fresh water. And so for fisheries, you can see about 11 metric tons from freshwater, but the majority of it, 80 metric tons, is coming from the sea. And then you can see for aquaculture, there's more seafood or fish coming from or being produced that's freshwater based, such as tilapia and catfish, striped bass. And then there's less in the marine area. And this is because there's a lot of species fished in the ocean that can't necessarily be grown in production yet because it takes a long time to understand the biology and then to be able to actually culture it in a way that's sustainable. And so I want you to keep that in mind from a biodiversity standpoint. But you can see right now aquaculture is about 50-50 coming from the sea and coming from aquaculture production. Okay, so since aquaculture is playing such a big role to help aquaculture and fisheries for that matter, to eliminate hunger, to promote healthy lifestyle, to reduce poverty, and also have a big impact on employment, what we're seeing is that there needs to be an increase. And so the name of this session was to feed the 10 billion. And so if you look here at this graph with the million of tons of seafood along here, here we are in the 1960s, 1970s. 1970s is when I first started working in the field of aquaculture. So it was just kind of a glimmer at the time. And now here we are, this is 2010, and you've got 58% of the seafood coming from fisheries, 42% coming from aquaculture. If we fast forward ahead to 2050, which is close to 9.7 billion people expected in the world, you can actually see a 10% increase or even more so of fisheries. It's already reached a plateau and now it's starting to go down. But you can see this incredible soar of the need for aquaculture. And this is why I say that aquaculture is one of the saviors for a protein source, a healthy protein source for the growing population. Aquaculture is right now outpacing world population growth, and it is the fastest food producing sector. Whether it will be able to stay ahead of that, it's really, we need to wait and see. But there's lots of technology in place, and we'll talk about some of that. With all the urbanization that's going on, especially in developing countries, population increasing and more middle class that can afford seafood, we're going to see even more. And so I wouldn't be surprised if this projection is actually low. So where is all the aquaculture coming from? And I really like this slide. This was given to me by a colleague, and you can see that the world is skewed according to the amount of aquaculture that's being produced. So you can see that the Asian countries are 91% of where most of aquaculture is coming from in the world. And so there's certainly some being produced in the United States, but it's a very, very small amount compared to the rest of the world. So speaking about the U.S. aquaculture species that are fish, I thought I would show you a few of those species here. How many of you eat aquacultured seafood? So do you eat some of these species that are up here, like salmon, for instance, and trout, catfish, tilapia, hybrid striped bass, shrimp? There's quite a variety of different shrimp species cultured in the United States: crawfish, clams, oysters, mussels, and also seaweeds. These are some also very popular fishery species that are, or aquaculture species that are grown around the world. So we come back to the question, how will aquaculture be a savior to help feed 10 billion? I first want to start off with just talking about some of the challenges, but also opportunities that aquaculture has to face to be able to meet this growing demand for seafood. And so if you look at the first one, which is something I already spoke about, food for more developing countries, and there is a lot of work that's being done overseas to help developing countries to be able to grow seafood in their villages and in their own backyard. So production technology, I'm going to talk with you about this. This is very important because of the cost of seafood. You need to be able to produce seafood at a cost-effective method, but also to save on resources at the same time. Alternatives to feed ingredients. There's quite a bit of fish meal and fish oil that's used in fish feeds to feed aquaculture. So there's a tremendous amount of studies going on in our aquaculture field to look for alternatives to fish meal and fish oil, and also to be able to use fisheries byproducts to be able to refeed to the fish, to look for poultry byproducts, to look for soybeans and other legumes, to also look for algae production and plant production. And so you can see this is where the two meet, all the discussions that we've been having today with all the plants and the collections of algae and the necessary plant products are needed also to help aquaculture. And so we also need to make sure that we maintain biological and genetic diversity, and then also overcoming some of the stressors such as climate change and ocean acidification. We're seeing a lot of that with our shellfish production, where the oceans are becoming acidic, especially on the west coast of the United States. And so this is very harmful for the shellfish that are being grown there. So I'd like to introduce you to Harbor Branch Oceanographic Institute. We are located in Fort Pierce, Florida, and which is on the east coast of Florida. We have an aquaculture park, we call it. It's about 30 acres in size. And I've been working there for the last 20 years, and we do a lot of development of technology that can be used in industry. So what you see here are many what we would call greenhouses, which I think is the term that you all use for plants as well. Inside the greenhouses, they're full of tanks, and we're working with fish and shrimp and shellfish and seaweeds, and we're looking at different production technologies. We're very well known for what we call recirculating aquaculture systems, which allows you to reuse your water. It allows you to grow your product year-round indoors. It reduces water consumption because you're reusing the water. It also helps with effluent discharge, so you don't have much waste products because you end up reusing that water again. It allows for intensive production of seafood and enhanced biosecurity, and also increase food safety because you really do have control over what you're growing in these tanks. We have a new, I suppose a new technology. It's actually being used around the world. It's called Integrated Multi-Trophic Aquaculture, or IMTA. And this is where you not only have one crop that you're growing in your system, but you have multiple crops growing in your system. And so the waste from one product is actually a resource or a food for another product. And so you're actually helping to save on waste, essentially. So how does that work? Well, here's a very simple diagram. And I like to explain it as having like a bicycle wheel. And so in the middle here is your filtration system, so the hub of the bicycle. And so you have over here your shrimp and you have your fish. And they're both fed a feed. And then they have some waste products. They don't always eat all the food. They do practically eat it all. But they do have waste products that are bioflocculated, and the bacteria makes a really great food, not only to feed more of the shrimp, but if you take the filter system, it particulates it out, and it can be fed particles to the oysters. Grazers like sea cucumbers or urchins can graze along the bottom. And then the micronutrients, the nitrates and the phosphates, can be taken up by the seaweeds, by the microalgae, and then in turn be fed back into the system. Working on this now for about we're on our sixth year of working with the IMTA, and we're really finding that it has great value in growing multiple species in a small footprint, but at the same time being able to have a situation where you can have all different levels of the food web, which allows you to see a nice ecosystem within your aquaculture facility. So I want to switch gears for a minute and talk about offshore aquaculture. And this is farming fish and shellfish and also seaweeds in the deep ocean, in open marine waters, in usually areas of strong current. Some of you might know that NOAA, the National Oceanographic Atmospheric Administration, aquaculture and fisheries did release a ruling to be able to have more offshore aquaculture, especially in the Gulf of Mexico. So some of you are probably very familiar with the fact that a lot of salmon is grown in net pans. In fact, about 99% of the salmon that we eat today is from aquaculture, and it's mostly grown in these sea farms and in Norway and Chile and Canada and also the US and Scotland. So this concept of marine aquaculture is actually all around the world, and you can see quite a number of different places where it's used to grow fish and also shellfish and also oysters and mussels for shellfish and seaweeds as well. So I wanted to bring in the concept of collections and how important they are also for aquaculture. And I talked a little bit about the need for agriculture-based feeds to help to feed fisheries. We also have heard about different phytoplankton collections. Genetic selection of broodstock is very important. Cryopreservation of sperm and germplasm program. And we've also touched on this during this symposium about the National Animal Germplasm Program in Fort Collins. And there are actually fish and shellfish that are stored in this area. So looking ahead to the future, this is what we might look like. A very integrated system with our offshore cage cultures, our integrated aquaculture systems on land, helping to grow seed stock that can be put into the cages. And then I really like this one because it combines not only the land, but also the sea. And so the sea is such an important part of our life here. So the next time so that's the future. The next time you're at a seafood place, remember to buy sustainable seafood, both from the wild fishery stock and also from aquaculture. And if you need some help, there's lots of seafood guides out there. The one that I like the most is Seafood Watch, put out by Monterey Bay. And it tells you what are the best choices, the okay choices, and the don't buy choices. I have a few people to acknowledge that helped me with this, providing a few slides for this presentation, and so you can see them there. And then I just wanted to end by this wonderful passage that is from the FOA report. Promoting responsible and sustainable fisheries and aquaculture is key to the health of our planet. Our own health and future food security all hinge on how we treat the blue world. So thank you. I do have, I think, a few minutes for questions, do I? Right, so farm salmon, there are some very good sustainable practices for farm salmon. And so not all practices are equal. It depends on the company. For instance, I know some people, or not some people, I don't know them personally, but I know some companies in Scotland that do a really good job of farming salmon. They take into consideration the type of feed they use. They do fallow work where they've had fish in a cage and then they'll let it sit dormant for a period of time, then move the fish to another area. So you want to look for sustainable practices in aquaculture. It's not the same everywhere, just like fisheries is not the same everywhere either. I think there might be an extra mic here. Okay. Okay. The question is about genetic stock, and it's a great question. And although aquaculture is a very old practice, that you can also see from more from a sustainable or more from a backyard subsistent farming practices, but really we're a very young industry in many ways, because you can see from the graph that really it wasn't until the 80s and 90s and now in the 2000s that it's taken off. So a lot of companies have their own genetic strains, their selective breeds, and they have hatcheries, and then they sell their seed stock to other farms to grow them. And so that's part of their proprietary part of their business. So it's not like you can just go and buy certain seed stocks. In some cases you can. In some cases they'll sell you brood stock that have certain genetic lines. So I would say yes, but I think there's a lot of room for improvement there. So you're going to get some of that, but not all around. Yeah, no, it's a really great question. I'd like to look into that a little more too. I would have to look into that. Great question. Yeah. Yes. That's true. That's true. Yeah, there is some traceability of the seafood, both from the wild and also from aquaculture in terms of where it's coming from. But actually the gene tracing. Now, one thing I want to let you know that in aquaculture, it's genetic selection of breeding. It's not changing of the genetics. Like there's been a lot of discussion about the frankenfish and the change in the genes to get faster growing salmon. It has not been accepted. So everything's been done through selective breeding only. Okay, very good. Thank you also.
The reason why I have notes is I know I have too much to say, so I'm going to try to stay brief. What's that? Questions at the end? Yeah. Like Rod Page, my contribution to this discussion is primarily on the informatics. But unlike most of you, I'm situated at a library, not at a museum or a research center. And our library here at USDA is situated within the Agricultural Research Service, which currently has a grand challenge. And this is a challenge to the researchers to do what they need to do to create the knowledge that will transform agriculture to deliver a 20% increase in quality production with a 20% lower environmental impact by 2025. Now, this is a tall order. I don't know that anybody really expects that we will achieve this, but it's the goal that we're currently shooting for. And so most of the researchers in ARS have been asked to think about their research in the context of this very food security-driven grand challenge. So I don't know how many of you are familiar with this book by Randy Olson. It's called Houston, We Have a Narrative. And this is a guy who started in biology and is now in Hollywood making movies. And so he's taken ideas from screenwriting to help scientists tell the story about their research in an engaging way. So here's my story. Currently, here in the library, we are making USDA-funded data open, discoverable, and safe. So this is the Ag Data Commons. I'll tell you a little bit about that project. Second, We're inventorying biological collections and their identifiers. This is the U.S. Federal Scientific Collections Registry, which David Schindel talked about yesterday. And then finally, I'll be talking about a project with the University of Maryland where we're adding semantics to open data in order to promote synthesis. So these are the and parts of our story. We're doing this and this and this. And the plot twist is, but we're not there yet. These data systems, none of them really are as mature as they could be. They're not well populated, they're not well linked, and they're not sustainable yet. And so the end of the story that I'll leave you with is the challenges and what we need to do to get on a better footing, and I'll get to that at the end. Okay, so the Ag Data Commons is my primary project here at the library, and this is designed to be a place where ARS researchers in particular can park their data safely, or they can point to where it might exist in some other trusted data repository. We cover everything from environmental measurement data, from the Long-Term Agroecosystem Research Initiative, to the genomics of livestock pests, to data sets related to modeling soil erosion, and so on and so on. So I think you're getting the idea that this is a very general repository, all for things that are relevant to agricultural research. So we're currently in a pilot phase. For the last year, we've allowed people to register for their own accounts and upload their data. We have about a little over 200 publicly available data sets right now, of which a little bit more than 100 are harvested from some other systems. So again, the idea is not that we have necessarily all the data in the Ag Data Commons, but we know where the data happen to live. We're set up to do distributed curation. So we're trying to be scalable. We're not going to have enough staff here in the library to review all of this incoming data that we expect and make sure that it's well described. So we're trying to set up so that there will be a level of curation around either USDA or beyond USDA that will help us make sure that what we have is good. We have links to PubAg, which is the library's or USDA's answer to publicly accessible scholarly literature, full text for those things that are created by USDA researchers. And we also tag the data sets with NAL thesaurus terms. So we have a thesaurus here. I'll talk a little bit later about it at the end. But this is basically some vocabulary that's specific to agriculture, but it does cross over into the biodiversity and other domains. We give our data sets digital object identifiers, which makes them easily citable and trackable. We use ORCIDs for authors so that we can disambiguate names by using these permanent identifiers, which are starting to become quite popular in the scientific community. And we use fundref codes, which the publishers are using to identify what the sources of funding are for these data sets. In addition, we have added ways to capture the metadata about the methods that were used to create the data set. And we do that because we're not just trying to meet the minimum level of compliance. Yes, the data is publicly available. We want to make it as reusable as possible. And finally, we're designing this to feed these scientific data sets up to data.gov, which is the federal government's access point for openly available data. Now, this is the machine readable part. On the left, you see a screenshot of a current data set web page. It happens to be the web page for the Germplasm Resources Information Network, GRIN. You can see there's a lot of text there that's human readable. We have links to the files and the web pages that GRIN has made available. We have some more structured metadata down here in a table in the bottom. But the important part is that this is available to other machines in the form of RDF and JSON machine readable code, which I'm not going to show you because it's not for humans, it's for machines. In addition, we are encouraging the use of data dictionaries, hopefully in a machine readable form. So basically, what these data sets have inside them needs to be documented in a machine readable way. And so we've got placeholders for these. Not a lot of our data sets have them yet, but we're hoping to improve the situation. And then finally, we're trying to make sure that all of those files, as many as possible, are available in machine readable forms, not as PDFs, not as images. We're also making sure that there are links to the various code that's used to create the data sets. and pointers to databases, and maybe even an archive of a web database that we can actually make easily downloadable on our site. Okay, so I'm going to switch gears now and talk a little bit about the Interagency Working Group on Scientific Collections that Dave Schindel told us about yesterday. Here at the library, we're hosting the clearinghouse of the documents for this interagency working group. So the reports and the policies that each of the U.S. federal government agencies have developed about scientific collections. And if you click on the link on the right, you go to the registry, which is hosted at the Smithsonian Institution. And we here spent the summer trying to populate this registry with information about the scientific collections that are held by ARS. And so, let's see. And as David said, the entire scientific collections world ranges from microbes to moon rocks. Here at ARS, we're focusing more on the biological collections. And this is, you probably can't read this at all, but this is what you get if you search for GRIN, the Germplasm Resources Information Network. It's actually composed of a number of different locations, each of which specializes in a different kind of germplasm. And so we have not only the institution code for the overall thing, the institution that they're within, but also the specific codes that are used for each of those different collections. And so the value here, what assists machine readability, is that these codes now are things that we could use in a controlled vocabulary to do some text mining, to find the literature, to find the data sets that are related to these resources. All right, we're still in the process of adding data to that. Just in this meeting, I've gone through several of the collections that have been talked about already to make sure that we have them in here and to make sure that they're correct. All right, so those are the identifiers. Okay, now I did a quick and dirty analysis of what we have so far towards machine readability. And let me just explain that this is the overall number of collections. It's a little bit under 80 collections that ARS holds. And of those collections, a little more than half of them are called institutional collections. Those are the ones that have some sort of ability for other researchers to use and that have formal accession projects. The rest of these are project collections. They're important too, but they're not usually accessible outside of a research project. They're probably destined eventually to become part of the institutional collections, but we're inventorying them now just so that we can have a sense of what's actually out there in USDA. Now, if you take this institutional collection bar and look at how many of them actually have an online catalog, the picture gets a lot worse. So less than half of these institutional collections, at least that we're aware of, have online digitized catalogs. The rest of them, even though they're institutional, don't seem to be available online. So there's no data about them that people could look at. Now, I may be wrong. We're still trying to clean up the data. But then again, if you look at the ones that do have online catalogs and look at which of those have APIs, application programming interfaces, things that developers could use to write programs to make use of the information. And as far as I know, none of them have APIs yet. Now, GRIN, from what I understand, they do have APIs that they use internally, but those aren't publicly available APIs. So I think what this tells us is that there's great potential. There's a lot of collections that are managed by ARS. There's also collections that ARS contributes to that are held by the Smithsonian. They're not included in here. So there's definitely work we can do to improve the machine readability of the information in the collections that ARS manages. Okay, so the third project I want to tell you about is a collaboration with University of Michigan's Socio-Environmental Synthesis Center, SESYNC. They're based in Annapolis, and they're called Data to Motivate Synthesis. And they have taken 300 public data sets. Those are the ones that are in data.gov that are related to agriculture, energy, or water. And they're doing work to see the feasibility of integrating all of these data sets. And so they pulled out 200 measurements that are in those 300 public data sets. And they're doing some ontology linking to see how do these data sets connect. And so what they're trying to do, you probably can't read this, but they want to go from the classic data-driven workflow where you get a team of people together, you talk things over, you think about questions, and then you go look for the data and then you analyze the data. Instead, what they want to do is have people start with the data that's actually out there. Don't waste your time trying to talk about things that you're never going to be able to get access to. Start with the data, use that to generate the kinds of questions that could be answered with that data, and then maybe put together the right team of people to ask and answer those questions. So I'm not going to go into too much detail about this. Their work is still in progress, but as an example, they've actually put the GRIN database into their system and spelled out some of the measurements that are specific to GRIN, and they put it in a larger context called the Food, Energy, Water Nexus Framework, which has water systems, food systems, and energy systems. And they've mapped all of these hundreds of data sets to these concepts. And so you can see here seeds, varietal characteristics by group, seed operating costs. So they have various concepts that are in these data sets. And the idea is that researchers can browse around, look for related concepts, and then actually download the data that is available to answer the questions relating to those concepts. Okay, so now we're getting to the plot twist. It's still early. What we really want to do is help answer those questions. In order to support those revolutionary advances in food security research, we're going to have to do some work to make these systems more populated with good data, linking across these systems or sustainable. And so, for example, we're planning to scale the Ag Data Commons up to hold huge numbers of well-documented machine-readable data sets. But that's a lot of effort, especially if you're doing it manually, which is a lot of what we're doing right now. And we don't have the automated processes we need to just automatically get it out of the literature. So for example, there's dozens, if not hundreds, of publicly available PDFs that have the results of performance trials of different varieties of corn and soybeans. This is just from last year. So these are reports that come out every year. Different states do them. We have paper copies up here in the library. I have been offered these PDFs as data sets for the Ag Data Commons, and I ran into a conundrum. Well, they're PDFs. I don't think of PDFs as data. Now, within those documents are things like, whoops, these are all the names of these hybrid corn versions, and then the basic yield information, moisture percentage. I'm not even sure what that is, and I don't know what that is either. But these are all characteristics that somebody has determined for these particular hybrid corn things. I don't know if any of these have vouchers in any of the germplasms. I tried looking up some of these sort of cryptic looking identifiers. They don't seem to be in GRIN. So they're not connected to anything. And anyway, these are still all in PDFs. And so we have some work to do to get the data out of these kinds of reports and into a machine readable form that we can do something with. All right. We also need to increase our use of identifiers, both in the Ag Data Commons and in the U.S. Federal Scientific Collections Registry, so that people can do the kind of automated mining that Rod was talking about. If we get these identifiers populated, then we can start looking for them in the published literature. Now, the Data to Motivate Synthesis work is a research effort that is tied to a synthesis center that may not have a lot of longevity. Many of the work that we're talking about here, some of it's being done on soft money, research money. At the end of the grant, it may go away. What's going to happen to it? So we want to capture the results of the work, particularly that the Data to Motivate Synthesis effort is doing, to semanticize those data sets that are public. So they've already done some work to connect the dots across different kinds of data set. Let's get that information, whoops, go back, not there yet. Let's get that information captured in the Ag Data Commons, so that if the Data to Motivate Synthesis Center effort goes away, at least the work that they've done is still available. But furthermore, we need to connect those terms that they've used to existing ontologies that are out there. So this is a screenshot of the Global Agricultural Concept Schema, which is actually a merged vocabulary of 3 different efforts that are capturing a lot of the terminology that's used in agriculture. And so we have that. Okay, just a couple more and then I'll be done. So even before we've fully populated or linked the data, we need case studies. And I think we've talked about a bunch of them here. But we basically need to combine the data in new and innovative ways, in ways that will actually help people. And this is part of the example of making the long tail small data large by combining it together. And one example of that is the agricultural model intercomparison and improvement project that is taking a lot of small scale crop studies, putting them in a giant database, and using them to model what's going to happen to crops under conditions of climate change. And so one of the things that came out of that AgNIP effort that I just talked about was a study that's pre-published on agricultural systems, prioritizing climate-smart livestock technologies in rural Tanzania, a minimum data approach. And so they're starting to become more and more data-intensive studies that are not necessarily going out to the field and measuring new things, but they're taking the data that's already out there to make some predictions and put that data to good use. Okay. So the last thing I want to talk about is that in order to do all of this, we have to come up with ways to partner in both short-term agile ways and also long-term sustainable ways. And so some of the things I've talked about are from academia. All of Rod's stuff is great, but what happens when Rod retires? What's going to happen to all of the great tools he's built? He is not going to be able to keep them up forever. So the work with University of Maryland that we're doing, the work with AgNIP, all of that is funded on short-term funding. So the role of the government agencies like USDA may be to provide the long-term storage, maybe the cloud storage that at least can capture the data that's resulting from those efforts. And then also collaborating with these institutionally supported projects like BHL you've heard about and Encyclopedia of Life, so that that information can actually stick around beyond those short-term projects. And then finally, there's a role for some global organizations, particularly standards organizations like TADWIG, the Biodiversity Information Standards Group that I've worked a lot with, GBIF, which is the global repository for all the specimen data as well as occurrence data, and then Research Data Alliance is another effort that's really putting together good global best practices for this kind of stuff. Okay, and so with that, I'll say thank you. We wait for afterwards.
Yes, yeah, that's right. Yeah, but this is based on a paper that I did with Nature and Development Foundation outside material science, and I had it published with the 2015 FAO meeting in South Africa, and then hopefully it might be interesting to you. So the question is, how may resilient strategies and edaphic characteristics be associated? Implications for sustainable development goals and food security. So many ecosystems-based and community-based resilient strategies have been compiled in literature across agroecological zones and agroclimatic zones to inform sustainable development goals and food security programs. And popular ones are classified into biotechnological, ethnobotanical, biogeological, biophysical, sociocultural in nature. And these ones are well highlighted in literature, but not well so how common resilient strategies may be intricately associated with edaphic characteristics, soil characteristics. So that's this. This is a desktop review that attempted to establish any associations that might exist between resilient strategies and then edaphic characteristics. So what I did was do a desktop review of all the resilience strategies within the agroecological zones in Ghana, and then try and see if some of them were tied to certain certain locations, certain soils. So the finding that came out was that common resilience strategies appear to belong to certain localities. And then actually they were actually soil series specific. So this suggests that common resilience strategies must be intricately associated with edaphic characteristics. And the edaphic characteristics must, in some way, naturally select resilience strategies. And in which case, edaphic characteristics must control the conception, creation, evolution, transfer of resilience strategies. So the loss of certain soils must translate into the loss of certain resilience strategies thereof. And any actions which seek to promote food security, sustainable development goals must seek to at least maintain local soil characteristics. And towards this, the youth would be very instrumental to sustainably grow biomass. And talking about biomass, insects and then grasses will be very key, because you can grow insects very fast. You can grow grass also very fast, because they use C3 photosynthesis and the likes for food, for fuel, for feed, and the likes. And then again here, pollinators and dispersers will also be very key, very, very important to this. And then I believe that there's a way that we can achieve food security and then grow biomass sustainably, and then pollinators sustainably and dispersers sustainably. So a periodic and exhaustive collection of resilience strategies, soils, insects, grasses, pollinators, dispersers, would be of key interest to the National Agricultural Library, and of course to Ghana also, and other parts. Thank you very much.
Good afternoon, everyone. Thank you for the organizer at USDA, also at the Smithsonian, a couple of also other, let's say, institutions in scientific collections, for sharing with you this afternoon a couple of, let's say, indication how the extent of collection can indeed serve, let's say, the purpose of food security. CIAT is part, just as Anne-Marie has indicated, part of the consultative group on international agricultural research. It was founded almost, let's say, 50 years ago. It started as a center on agricultural research for the tropics, however, let's say, relatively quickly, as the other sister center of the system. We devote ourselves, let's say, to the challenge of improving agriculture and food availability across the tropics and subtropics through plant breeding. And you know that in order to do that business of plant breeding, you need, let's say, access to the diversity, you need the traits, therefore you need germplasm, and you need to evaluate that germplasm. Here you have, let's say, the 10 of us, gene bank managers responsible for these gene banks. You can see where they are distributed and also the crops they are responsible for, basically staple food crops in the major families on which our food is based, as well as you can see, let's say right now, responsible for about 10% of the total accessions kept, let's say, worldwide in depositories, be seed depositories or tissue depositories for crop germplasm. Also, you can see there, let's say, that over these last 3 years for which we have records across, let's say, the 10 of us, we have distributed more than 300,000 samples. That is in the framework of our agreement that we have with the International Treaty on Plant Genetic Resources for Food and Agriculture, where it was rightly mentioned this morning, for about 50 crops, we can distribute materials relatively quickly once approved the material transfer agreement and once, let's say, plant quarantine issues are cleared. At CIAT, we are particularly responsible for, let's say, 3 groups of crops, beans, cassava, and tropical forages. You can see there, let's say, what we are keeping in terms of numbers of accessions, as well as the number of countries providing those germplasm since the beginning of our work. So this means, let's say, as you can see, something close to 68,000 items of diversity in these 3 groups of crops, representing the heritage of a bit more than 140 countries. And that is, let's say, from small herbs like, let's say, the zornia or the wild relative of Arachis, plants, let's say, 1 centimeter tall, up to multi-purpose trees. We are, let's say, responsible for that conservation. I used to say, let's say, that I have a few countries helping me to sustain the lab and the 4 stations that we are operating in Colombia, provided that we can distribute. We can distribute germplasm. This afternoon, I don't know what, let's say, a user somewhere on the planet may ask, and therefore this means that the almost 68,000 items of diversity are available at any time for those purposes of food and agriculture. You can see that in the history of the lab, we have distributed more than 500,000 samples to users in a bit more than 160 countries over that period of time. Now, quickly, let's say, going through the 3 collections. In beans, of course, let's say, and it was, let's say, the initial purpose. The main user is, let's say, the programs that we have at headquarters and across, let's say, the regions, in the case of beans, particularly, let's say, Africa. But also you can see that agricultural research services of countries as well as university departments are also important users of those collections. An important point also is to see that indeed we have distributed almost entirely the collection that we are keeping. With the forage and for the seed germplasm of forages, this is a slightly different situation, as you can see, where, let's say, increasingly, and this is because we have concentrated on the improvement of few brachiaria and panicum, let's say, grasses, the external user are becoming, let's say, more important as compared to the internal user. Here we have made less use of the collection. I will, however, let's say, show you a few examples where indeed, let's say, the use is very different from what we expect at the beginning. And just, let's say, to reinforce a point that was made by one of the former speakers this afternoon, some of the germplasm that we are keeping initially for feeding cattle is right now used, let's say, elsewhere for feeding, let's say, farm fish. Cassava, although it is distributed, let's say, as tissue and using, let's say, the in vitro technology, again, you can see that we have distributed almost the entire collection. We have distributed almost, let's say, 5 or 6 times, let's say, and the collection coming from 28 countries, we have distributed that to 84 countries across, let's say, the tropics, as well as labs, for instance, in the US, in Europe, or as far as Australia. Now, one example is, let's say, how, let's say, the collection, in the case with the bean, has been able, let's say, to respond to breeders across time. At the beginning, and it was, let's say, logical, it was expected, let's say, it was, let's say, to make sure that farms are harvesting something. And to do that, let's say, plant breeders are then, let's say, transferring resistance to disease and pest. The next step is, let's say, then the concern on yield. And as you can see, then it was yield, let's say, but let's say in very poor soil. More recently, we have, let's say, from breeders, a request for nutritional quality. And we can see over the last 5 years that we have request of germplasm in order to challenge the problems related to climate change, such as, let's say, heat, drought, or flooding. Now, for some of you, let's say, in the back, I will not pass an exam, let's say, quoting what you can see, but the message with this is a compilation where, let's say, you can see a particular gene linked to a particular accession of the gene bank, let's say, and when that work was published, it's, let's say, indeed quite an impressive, let's say, time spent. Is also, let's say, a very huge diversity that has been used. That is, let's say, from land races to, let's say, crop wild relatives in the primary and the secondary gene pool, as you can see. And again, let's say, for all diseases and pests affecting that crop. It is also, let's say, for other, let's say, such as, let's say, abiotic stressors, also nitrogen fixation, yield, and nutrition, sometimes, let's say, with things relatively unexpected, such as, let's say, the use of, let's say, of wild relatives to exploit, let's say, a quantity trait loci for, let's say, the increase of yield, but also to use wild relative, like, let's say, for instance, yes, wild common bean for the improvement of, let's say, amino acid contents, let's say, in seed storage proteins. I will go through very quickly a few examples, also showing, let's say, how those collections have been used also specifically for more, let's say, research purposes. Of course, let's say you have had the case of the use of accession straight, let's say, into national catalog of plant varieties. You have had, let's say, the case of that tropical grass, Brachiaria, released in Brazil and planted over thousands of hectares. Also, one particular accession from Colombia of Styloscentis guianensis, planted on 1 million hectares, let's say, in southern China. In breeding, you have just seen, let's say, the diversity of use. We have, for instance, okay, one land races from eastern Ecuador being in the cassava collection out of, let's say, the more 5,000 something land races, only one resistant to whitefly. Better for us, let's say, that we are not losing it, let's say, throughout, let's say, the maintenance of the collection. I've been myself, let's say, responsible for a couple of, let's say, new bean species, not only sending types to institutions such as, let's say, the Smithsonian or Kew or Missouri Botanical Garden, but also keeping seed so that, let's say, people in the future can also, let's say, do additional, let's say, botanical or taxonomical research on those materials. Here, let's say at Bellsville, a colleague pathologist has been asking, let's say, that particular material for leakage mapping of loci for the resistance to most important diseases, let's say, such as anthracnose or angular leaf spot. Our colleagues in Montpellier have been asking, let's say, our entire collection of Echinopsis over the last few years, and that is because it's, let's say, a very interesting tropical legume where, in addition to nodules on the root system, you have also nodules, let's say, in the aerial part on the stems. And that particular one, let's say, Echinopsis evenia, was particularly key for the advance of that research. We have also, with cassava, something interesting that is, let's say, called the post-harvest physiological deterioration. It is almost extinct in Texas. It is, let's say, Maniote walkeri in the San Antonio area. That is also, let's say, while the species interesting for that trait, but that particular material from Brazil is also particularly relevant. A couple of years ago, Dolores Piperno, working at the Smithsonian in Panama, she asked me, let's say, different materials of common and lima beans. And why? Because she needed that, let's say, for her archaeological studies. She was, let's say, studying ancient diet of people, let's say, on the Peruvian coast. And therefore, let's say, modern accessions, what, let's say, particularly to help her, let's say, to identify, for instance, the different sorts of starch that she was picking up, let's say, in the teeth of, let's say, of archaeological, let's say, remains. Also, let's say, accessions have been very useful to advance, let's say, genetics. For instance, Mark Bassett at the University of Florida was using, let's say, to understand more the genetics of, let's say, of seed color and seed patterns. Also, for instance, here a case with, let's say, a dominant mutation in leaf development. And more recently also, it was the first true genetic map published, let's say, on the common bean, where land races from Peru has been used, let's say, for that reference genome. Concluding remarks, it is, let's say, beyond numbers, I think, let's say, the diversity of the collection that allowed so many, let's say, colleagues to advance, let's say, in their own work, and I think that is, let's say, the way it should be. Of course, let's say, and it is particularly true, I believe, let's say, for the forage collection, I continue to see that it's what I should call, let's say, a sort of regional collections. And what I mean is I continue to wonder how far, let's say, people know about, let's say, those collections and about the potential. And particularly, I'm referring to extensionists, for instance, in Africa or in Asia. That is then, let's say, for us, let's say, a sort of call of attention to think a little bit, let's say, when we are designing, let's say, our database and our website. And that is, let's say, to change a little bit, let's say, the chip beyond, let's say, what I would call, let's say, curatorship. Of course, let's say, you need a lot of data, very accurate data, for the maintenance of your collection. However, let's say, this is not always of straight interest, let's say, for people, let's say, in agronomy or farms and so on. And therefore, let's say, you need, let's say, to rethink carefully, let's say, how you are presenting, let's say, your database and your website. In relation to this, let's say, we are joining, let's say, with our colleagues, let's say, of CIMMYT and CIP, these two centers, let's say, in the Americas, let's say, to adopt, let's say, Green Global, but that is with the view of developing a community of practice. One important element in this is to use, let's say, mobile devices, such as, let's say, tablet, to capture, let's say, data both in the labs and in the fields. Also, an interesting development that we are doing right now is to recheck all geographic coordinates of landrace and crop file relatives, let's say, because of the numerous GIS application that we can see right now in those communes. We did, let's say, in the 90s on purpose a decision that we would not welcome too much segregance of breeding programs, but rather, let's say, to focus on what I call primary source of variation. And that was a specific search for land races as well as, let's say, crop wild relatives. At that time, it was not, let's say, with the level of urgency that we have seen, let's say, over the last 5 years. This is where, let's say, a vibrant community of scientists, and particularly, let's say, we are very fortunate, let's say, to have that for beans and for cassava. There is also, let's say, helping the gene bank, and that is, let's say, to provide, let's say, with indication about, let's say, the sort of traits they are working on, the sorts of germplasm that they need, and that is very important for us, let's say, in order to have, let's say, a sound acquisition of new germplasm. It should not be, let's say, a collection of the past. We should, let's say, be able, let's say, to advance, to continue to welcome material that is not present, but that should be done, let's say, with some good feedback, let's say, from those communities. I used to say that food security starts with the right name, down to the gene, and then back, let's say, to the phenotype. This means, let's say, that for us, it's a sort of, let's say, approach as a multi-service. We have, and particularly for the foragers, let's say, a digital herbarium being developed. We are, let's say, progressively preparing, let's say, to put sequence information, but also, let's say, trait documentation. And that is basically, let's say, to link accessions with, let's say, true publication, with traits, let's say, that have been, let's say, disclosed, let's say, by other people. A current challenge for us, let's say, and towards the future, I believe, let's say, is, let's say, to find people with the sort of, let's say, multidisciplinary education, that sort of focus, so that, let's say, we are able, let's say, to continue to serve as many actors in a global society as I think we should. Thank you very much for your attention. As a germplasm manager, let's say, you are always, let's say, having, let's say, some discussion, let's say, with your finance director. And therefore, let's say, that is back, you have, let's say, a certain number of operations to perform, let's say, on an accession at a given time. And therefore, let's say, I think it's so important that a particular accession capture a fraction of the diversity of that crop in such a way that right now, or let's say in a relatively predictable future, one user, let's say, will make something great with that particular accession. And that is then, let's say, I mean, that responsibility on us, let's say, to track down, let's say, what we call internal copies, to reduce internal copies. Yeah, to reduce, let's say, our redundancy. We have done that, let's say, for our cassava. I've mentioned, let's say, it is maintained in vitro, and therefore the cost per accession is the highest as compared, let's say, to seed materials. And fortunately, let's say, with markers, what we have done so far, let's say, it is not yet, it is, let's say, within, let's say, these 12 months, let's say, and it's not yet fully, let's say, on the entire collection of, let's say, the 6,000, let's say, cassava materials, but that is coming. But out of what we have done already, because those were collected before the distribution of elite improved varieties and in very different remote place of tropical America, those were therefore, let's say, relatively unique to these different areas and therefore, let's say, with a low level of redundancy. So that's, let's say, I don't have, let's say, a sort of final formula on that. It's more, let's say, a sort of, let's say, listening attitude towards, let's say, colleagues, analysis of trends. I have no regret to have collected bean samples is what is today Mexico City. Because I think, let's say, for Mexico, as well as, let's say, for the bean community, those will represent something useful in the future. But again, you need, let's say, a sort of continuing, let's say, discussion on that, because on the other hand, your collection can simply not grow forever, as you would have, let's say, some sort of unlimited resources. That would be unrealistic. And therefore, it would be then, let's say, somehow counterproductive towards, let's say, the goals that you want to achieve.
So I have a different perspective from a model organism perspective. And the two things I want to mention are, we have always had an archival category in the collection, but because we're so small, 2 people, it takes more effort to go and find something and remove it from the collection than it does to leave it. So something becomes archival if nobody's ordered it for 10 years. And that means it doesn't get subject to our routine periodic quality control. So we test things for viability and typically for genotype. Because we leave it where it is, if somebody orders it, it comes out of the archival status and it's grown up, it's tested. Unlike many collections, when we distribute things, we grow them up, we send people a living culture. And I call that arm's length taxonomy. We grow it up. Is it orange and fuzzy? That means it's Neurospora. So we've done 2 points of quality control. It's alive and it's what it's supposed to be at the first level of approximation. So that's the first thing. We have an archival status and our quality control is built in. And the second thing is about model organisms. So as a model organism collection, we have accumulated resources from other model organisms. So there is a kind of an arc of a model organism. They become developed typically to answer a specific question, because they have utility to answer that question. And then as research moves on, that particular organism may not be used anymore. So for example, in the 1920s and 30s, a lot of people used water molds because you could study mating. You could study the sexuality and the distribution or the inheritance of mating types. So we now have the world collection of water mold diversity and genetics. It consists of about 120 specimens in one refrigerator box. We inherited this in toto in 1985. Nobody asked for anything from that collection until about 2 years ago, when all of a sudden everybody wanted it. And we were very fortunate that they were preserved on filter paper, they're desiccated and kept cold. So we were able to share those at no cost with our research community. And people now have access to this archaic model organism that was all of a sudden of increased interest, both for biodiversity studies and because it's related to the organism that's causing the amphibian declines. So The value of the collection was that we were able to provide safe storage. We were able to advertise that we had it and make it available, even though we weren't actively curating this. So as a model organism, it had seen its heyday in the 30s, and it was resurrected again in the 2010s.
Well, I believe that if we could afford, we wouldn't want to lose any one single one of our resources, right? But they say that resources are limited in economic terms, right? I mean, if we could collect every single thing that we could, I mean, we'd want to keep them for the future because you never know. I mean, from anything biological, anything natural resource, if we could keep them, we had the resources to do that, it would be nice. But economically, I mean, we are a bit constrained, isn't it? But I believe that communities can play a role here, and social entrepreneurs can play a role here. It doesn't have to be government's responsibility alone. We could incentivize certain other actors to come into play to try and help out in this regard.
I know we have a question waiting here, but it's that idea, how could you incentivize private investors to invest in scientific collections? They do have to enterprise.
Yeah, Kev, you want to talk about the sustainability discussion?
To use his fantastic wealth to contribute to the global betterment of mankind. And at that time, he established the Carnegie Library System. And the Carnegie Library System bears his name, and it was a tremendous public good. There's still Carnegie Library buildings all over the US and in developing nations. I want to make the point that living collections are like modern libraries. And I've thought for a long time that we need global philanthropists to put their name on the living collection, a living collection resource around the world for the betterment of mankind.
I just wanted to add a little bit. Commercial companies do have interest in a lot of these collections. I mean, like Rich Humber's entomopathogenic fungal collection, most of the samples that he sends out go to industry, and they mine it for various chemicals and property. You know, they're looking for compounds. So I think that some of these government collections are used already by private enterprises. So it might not be that hard to get them to support them better.
Yeah, it generally is hard to get them to support things that are non-proprietary. Non-proprietary uses of this is support is very, very rare. The return on their investment has to be because they're going to get a major return. They're going to have to invest a lot in development. So they're not eager to give their money away and then have the information go into the public domain. And that's increasingly what I think we're discovering in our discussions about collections. And that is the relationship between, the trade-off between expanding collections and enriching the information connected to those collections, which is what makes this conversation so very interesting. CGIAR does not live on cost recovery of the exchange of material. It doesn't sell material, it exchanges material. That's the rules of the road there. And Kevin spoke to this before, that NSF requires cost recovery because they don't want to be supporting culture collections and stock centers in perpetuity. They want them to become independent and sustainable on their own. NIH has a different attitude. They say, we'll support it so that it will be distributed because we want to promote innovation. We want to make this material as universally available as possible. So now you have three completely different business models based on the funders, the benefactors. And we had this long and very interesting discussion in the last e-consultation. It's recorded for any of you to hear about these trade-offs. So the speakers said, they talked about biobanks, which are biomedical tissue samples, human tissue samples. They all start off just grabbing everything they can. They want to build the numbers because they're starting from zero. They want to have the maximum number of samples, the greatest diversity of samples, and people start to ask for them, and then they get paid for them. So that's sustainable in the beginning, but then they keep growing, and then suddenly the sale of the material doesn't keep pace. It's not paying for itself anymore. Now the bigger the collection gets, the more money they're losing. So they turn to other revenue streams. They start selling services. But CGIAR is in a very particular business of expanding the diversity of the cultivars they have while also gathering information about them, characterizing them and evaluating them. So there they have the trade-off of how much do they spend to add data versus how much do they spend to preserve things. In the absence of any revenue stream, they can get bigger and bigger and bigger. It's not going to make them any more money. They're depending on philanthropy and intergovernmental support. So I think we have to be specific in talking about these different strategies to what financial world do they live in. And there are at least 3 financial worlds that I know of, maybe even more. This trade-off, Daniel, between money spent characterizing and evaluating, what are the rules about the return of data and the release of data when you exchange material to growers and breeders, and they're going to do evaluation experiments on them? Can they keep that information secret, or do they have to return it to you?
Just let's say as USDA has done, let's say, for a long time. I remember, let's say, I've seen, let's say, in the past, those, let's say, yellow envelope with a seed, and there was a stamp indicating please return data to USDA. We continue to indicate that. I continue to ask, let's say, receptors of germplasm, could you please, let's say, share at least, for instance, the PDF or, let's say, or the reference about your work, where is that published, and so on, so that we can continue to link, because all donors are all the time, let's say, on our back asking us, but what is the impact of, let's say, of conserving that? It's not only, let's say, to show raw figures about distribution, where again we are somehow, let's say, sometimes in a sort of contradictory situation. Some pushing hard, let's say, you need to distribute more. Otherwise, let's say, this means that your collection are of no interest. But then, let's say, of course, let's say, if you distribute more, you need, let's say, to refill these seed stocks. Because it's not only, let's say, to distribute, it's, let's say, also, and related to that, the checking of the viability, the checking of absence of disease of quarantine importance, so that you can distribute safely worldwide these materials. Now you are pointing to something that is of concern to us, let's say, in a sort of, let's say, immediate and more long-term, let's say, and that is, let's say, the difficulty for us to continue to document further, let's say, about the useful traits. The sort of, let's say, golden 10 years at the beginning, where pathologists, virologists, entomologists were studying, let's say, all these, generating a lot of data with the possibility for us to collate those data, clean those, put the metadata. This is where, let's say, it is becoming increasingly difficult, and this is because of the structure of the funding, that we have moved from, let's say, a sort of core, let's say, and not really project-based, to to what we have seen over the last few years. That is project-based, 3 years, highly focused, and just that, and then end of story.
Yeah, in essence, you've moved from an institutional thinking, institutional collections, in a broad sense, the long-term investment of building information and collections, to a short-term one, like the project collections, which can be used up. You're not going to, nothing may outlive them. Yeah, the many regrets I have about college, I wish I had taken some economics, because modeling the economics of collections is real. We're missing. It's, Rod, I would put this in the category of things we know we don't know, because we think of the value of the collections is how many people ask for them, how many publications they produce, and we have no way of valuing the information attached to them. Because if we had a system of, if we had a marketplace where I'll give you a sample, but you tell me what you learn from it, and therefore the net value of that sample has gone up. It's, you know, if we were a business, we'd be looking at that bottom line, the value of the things and the value of knowledge, because the knowledge is now usable. But we don't have that mentality.
So along those lines, I was going to make a comment earlier that everything you say about the limited resources for physical collections is also true of data. So libraries don't just take things and keep them. They deaccession things, and they have policies about what is worth keeping. Now, it's a challenge with data that cannot be ever collected again because it was collected at some particular time and place, and conditions have changed. We'll never be able to go back and collect that data again. So there's some data that probably should not expire, and it's really hard to tell the difference. But I will say, last week I was at the International Data Week meetings in Denver, Colorado, and there were 2, maybe 3 entire sessions on the business models for data repositories. And emerging from those sessions was a call for new economic models for data. And it would be very interesting for those same people to do the analysis of, okay, what if the data is associated with something physical as well? So I don't think we're there yet, but I think a lot of people are talking about this, and I think the future is bright because so many people are talking about it, and we do have ways now to link across. So one thing that I would say is that the more that collections can share their data so that everybody knows what everybody else has, the more you can focus on those things that make your collection unique. Avoid the duplication of effort, share the, you know, if multiple collections are all collecting data about cassava, then make sure that all of the traits that are available for all cassava are available to everybody who has specimens. And so, again, it's sort of a communist view of, you know, it's the commons. Everybody shares what they know. The Global Agriculture Concepts Scheme that I mentioned before is actually a merging of 3 thesauri. One of the values of doing that was that each of those had a different strength. And one of those was in, for example, the different languages that things were translated into. So now, by putting it all together, they can actually have, I don't know how many languages, probably 20 or 30 languages that all of these terms are translated into. That's a great value. Interestingly, it's not, they didn't do this for all the concepts in all of these vocabularies. They took the 50,000 most used terms, actually maybe it's 10,000. They basically looked at what were the terms that were actually being used, and they merged those. So again, it's this trade-off between what's used and what's not. Now, for data, if something isn't getting used, we could put it into some cheaper form of storage and not look at it, very similar to what the fungal folks or the culture collection policies are. So again, it's a very parallel situation for what you do with data and what you do with physical specimens.
What role do you see of seed savers in all these collections? Because if you read Mother Earth's news, you'll see all these different, you know, places where you can send in to get seeds, and that as long as you agree to return seeds that they send you, you grow up the plant, return some of the seeds. And there's a vast network of that out there. How is the scientific community tapping into those different cultivars and the like?
My guess is that what's going to happen is parallel to what's happened with citizen science and observations, that over time those folk collections of seeds will become recognized, and there will be hopefully ways to deal with the quality control. I've heard criticisms of the seed libraries that some libraries, public libraries around the country, are starting to promote seed libraries, and some of them have been shut down because they're distributing things without a lot of information about what's actually in there, and you have to be really careful not to spread invasives, for example, that way. So there needs to be some recognition of, or pathogens or something like that. So there's this balance between, let's encourage people to do the crowdsourcing or the citizen stuff, but then let's also figure out ways to ensure the quality of what's being distributed.
Community. Home brewers are sharing strains of yeast very widely, and they may be microbial consortia. There's no quality control, and as yet nobody's been killed, but it's just a matter of time.
Is it saying the UK Plant Resources Group has a place for at least the big one, the Heritage Seed Library in Coventry?
So, Cindy, obviously I'm never going to retire. So it seems to me that every field I've sort of seen like this, there's a whole series of stages that we go through. And it seems to me that the libraries are probably the first. So when digital stuff happened, the first thing we did is we cataloged libraries. Here's a list of libraries, which is useful for libraries, but nobody else really. Then we digitized the card catalog, because that was fairly straightforward. And now we can kind of find the books, but that's not a huge advance. Then Google kind of blew the doors off and scanned the books. And the reason that was so important is basically you can blow the books apart. Now it doesn't matter the sort of packaging. You can sort of query across the books. And I guess an equivalent would be GenBank. GenBank doesn't care what genome the genes come from. You grab the genes, you play with the genes. So it seems to me that what we're looking for here is that kind of Google book scanning kind of thing. I think the more we obsess about packages and the data packages, which seems to be the big limitation, lots of data repositories at the moment, we're not going to get very far. We're going to have lists of data, which doesn't really do much. So if we follow that trend and sort of see what Google's doing, then I guess the next question is, what's Google doing now? And it seems to me it's doing two things. The first thing is it's not building these incredibly domain-specific ontologies, which we love. It's building a really, really simple one, which it's applying to a very simple ontology, very simple vocabulary, which they're using for medicine, for videos, for book reviews, for everything. So one approach might be, let's not do the domain-specific ontologies. I know we love doing it, but I seriously doubt it's worthwhile. The other thing, of course, they're doing is let's not do ontologies at all. We have text mining, we have deep learning, deep mind. So what happens when you unleash the artificial intelligence bots on this kind of data? What can you do? Google thinks they can do an extraordinary amount. So why aren't we thinking along those lines? It just seems that there's this constant series of steps we go through. And this community seems to be way back at the kind of, let's get the packages together kind of stage. What happens when we get beyond that?
So my response to that is, number one, you got to get it up digital in the first place. First of all, in any form, PDFs is better than nothing, but the more structured it can be, the better. And then, yeah, you can let Google go to town and maybe Google will solve all the problems. But having some, I'll call it seed money or seed starting is helpful. So the Google Knowledge Graph actually does leverage things like TraitBank, which has done some of the work to semanticize what's out there. So getting information structured enough that then Google can take it the rest of the way is very important. And so there's certainly people who argue that Google is the front end to all knowledge at this point, or whatever comes after Google will be. But but it first has to be out there to be found. And so I think, let's see, we should bring this back more to the physical collections, which is sort of the bread and butter of most of the people in this group. What's the representation of those physical collections that needs to be made digital so that some of this stuff can happen? I would argue that trait information is definitely going to be important to get out of these collections. The geospatial information that's already come up here as well. But also just the context, and the context, who knows what that's going to be. So that's the literature part, right? So we may, in our academic projects or in our small projects, each inch along a little bit towards one of these things. But ultimately, the more that's up there, the more that the AI folks can do what they do to knit it all together, I think the better we'll be. It's going to be a long time before you can actually ask the question, how do we solve the food security problem in a particular place, in a particular time? We're not smart enough to do that. So we're all just sort of incrementally trying to improve the situation. And so the Ag Data Commons, sometimes I think we're just way back in the basics because we're just putting up packages. We're putting up files and descriptions of files. But it's so hard to get all of the pieces together with a particular data package to really understand what's in it, that we have to start somewhere. The more we can automate that, the great if Google wants to take this on, but Google's not even really supporting Google Scholar. It's just a side project for them. So they're not going to put a lot of specific interest in the stuff that we care about. Well, with some exceptions, I think Google has sort of jumped into some public good projects, but we do what we do. We have to solve our immediate problems so that we can get the funding we need to keep getting stuff online.
So everything we do is with words. I mean, you can't, without words, we can't do anything. Physical objects are meaningless without words attached to them. I guess I'm not saying we should get Google to do it. I'm suggesting we should think like Google and adopt the same kind of approaches. So they basically, once you get beyond the packages, they don't care about them, they explode them, and that's when the really exciting things happen. And I think that's what you want to be heading towards.
And as far as you retiring, Rod, I'm less interested in whatever data you're generating, but in the tools that you're using to work with the data. So, you've got a lot of handy dandy things that most of our production quality systems don't yet take advantage of. And we need to make sure that those, what's going to happen to BioStore when you're no longer there to keep it running? So that there's a lot of functionality, I think, that we could be leveraging from these academic projects.
Sure. Thank you to our speakers.
Do you want to mention tomorrow? Or logistics for tomorrow. Starting the same time, so breakfast at 8:30. We'll get going at 9. And tomorrow is going to be a completely different format from today and yesterday.
Okay, so if you can refer to you may not have your agendas in front of you, but the breakout discussion groups tomorrow are free form in the sense that we'd like you to gravitate to one of 3 topics that is of greatest interest to you. And they are new strategies for increasing the use and impact of collections and their associated databases. So let's call that the strategies and impact group. Second one is case studies that exemplify this sort of cross-cutting use of collections in different disciplines and new uses of their data. And the third is to formulate major recommendations. So if you have some ideas for specific recommendations, gravitate to that group. So what we're going to do is ask each group to come out with a small number, let's say maybe 5 things that you think are most important. 5 case studies, 5 new strategies, 5 big recommendations. Can be more, but we would really like you to use the group to reach a consensus about this. This is where the symposium becomes a workshop. We really do want a product out of you, and it's the sort of thing that will go into this white paper that I keep talking about, to try to come up with recommendations and action items that we can put forward. They can be about funding, but please don't just say more funding. Be specific. What kind of funding? For what purpose? Coming from whom? If any of you have not seen it, actually, maybe we should, I've given Eileen a document to distribute to you. Who has not seen the green report about collections that the U.S. Interagency Working Group produced? Half of you. We'll distribute the electronic copy. This was generated in 2009. It was years of work surveying the federal government's collections. They came out with 7 recommendations, 3 of which have already been implemented. The clearinghouse and registry was one, that there should be explicit policies for managing federal collections. That's a work in progress. And there should be a system for establishing the cost of running these collections so that they should be budgeted for in the long term. And there's 4 other recommendations. It's got a lot of case studies. I keep harping on case studies, a lot of illustrations about impact of collections. So we'll distribute that electronically to you. We'd like to have something like that that's shorter, clearer, but points the way ahead on food security and coming out of this, something that focuses on food security. So tomorrow is boiling things down to be specific, and each group should look for a small number of deliverable recommendations, case studies, or strategies. That's what we're after tomorrow. And we're going to leave it to you to self-organize, aren't we? To pick a rapporteur who will report out.
If you didn't remember half of what, we'll repeat it. All right, thanks everyone.
Thank you all.